So I see that we have 24 out of the 28 students who originally booked tickets for this event are now registered and in attendance in the private Zoom webinar room. Welcome and thank you for being on time, everyone. So this is going to be the next in our series of advanced level biology revision webinars. In this webinar, we're going to be addressing the subject of genetics, populations, evolution, and ecosystems. And in keeping with the format of the previous webinars in this series, I have two Zoom webinar rooms, one of which is for the students at the LeSwap Consortium, and one of which is for the students who have registered and booked tickets privately at my website, invisible-college.com. And in keeping with the previous formats, only the chat box from the LeSwap Consortium students' Zoom webinar room will be displayed. As yet, they have not logged in, so as and when they log in, I will put that up on the screen. Now, again, in keeping with the format of the previous webinars, can you please make sure that any questions that arise, those of you who are in attendance, if you'd like to ask any questions, please don't put those into the chat box because that tends to scroll by pretty quickly and I might miss your questions in the course of the webinar. Instead, if you could use the Q&A box in your Zoom webinar interface, that way those questions will be held for the end of this webinar, which is when I'll address any questions that you guys have raised. So, as I say, this is going to be on the subject of genetics, population, evolution, and ecosystems. Now, this corresponds with section 3.7 of the AQA Advanced Level Biology Specification, which you're seeing here. Now, again, even though this is kind of generally geared towards students studying under this specification, the ideas and the topics that we're going to be reviewing in this webinar are equally applicable to those of you who are studying under OCR, EdXL and other exam boards for advanced level biology. So again, I'd encourage all of you in attendance to have your specification open to the relevant section. In this case, it's going to be this section here, 3.7. Let me just make that a bit bigger. Now, each of these topics is rather large, so I'm going to be going through it in as much detail as I possibly can. And again, just like the previous webinars in this series, if we do run over the 3-hour time limit, obviously not including the Q&A section at the end of the webinar, then what I will do is I will leave it up to you guys in the Zoom webinar rooms as to whether you want me to continue beyond the 3-hour limit and cover the remainder of the topics, or if you'd prefer me to upload an additional video to your private coaching areas on my website, invisible-college.com. Now, so we're going to be going through this topic pretty much in the same order of ideas as appears in the specification. So we're going to start with general kind of definitions concerning monohybrid inheritance and kind of introducing some key ideas in genetics. We're going to go on to study the idea of populations, and we will explore the Hardy-Weinberg equation and also move on to the idea of evolution and speciation, and we'll finish off with the ecology segment of this topic. Now, I must warn you in advance, this topic is one of the larger topics which I've created course modules for on my website. The course module on my website, I think, contains, let me just have a look here, I think it's about 16 lessons in the course module. Now, these course modules are designed to teach you everything that you need to know to get the very highest grades, A stars, from scratch, essentially. And it does go into a lot of depth here. Now, obviously, we're not going to be going into that sort of depth in the course of this webinar here. Instead, we're going to kind of skate over some of these topics and just provide a kind of revision overview. Now, if you are interested in exploring these topics in a lot more depth, there's somewhere in the region of 14 to 16 hours worth of teaching videos, hundreds of quiz questions and various other resources that you can find here. Now, I know that many of you who are in attendance at the moment already have access to this, so there's no need for me to go into quite as much depth as appears in this course module on the website. But as always, if you have any questions that arise after the webinar, then you can all obviously log into the website and into this course module and then leave your questions either as comments on the relevant lesson pages or as comments and questions in your private coaching area, which I will then address and answer there. So let's get started then. So we'll begin by kind of introducing some key ideas in genetics and then go on to talk about some more kind of advanced ideas here. So we'll begin with the idea of monohybrid inheritance here. So it all starts with Gregor Johann Mendel, and he was an Austrian monk who came to be known as the father of genetics because he really did most of the kind of foundational work which led to the modern science of genetics. Now, he formulated the so-called laws of heredity, which is really the basis for modern genetics. Now, during his time, it was generally believed that the traits or the characteristics of parents were somehow blended together at fertilization to provide the offspring with their characteristics. Now, it's important to remember that Mendel himself didn't really have any idea of what we call genes or the kind of molecular genetics that we all know about in the kind of modern science of biology. Instead, he did some very kind of more empirical kind of observational work by taking very simple plants like garden peas and then crossing those garden peas with very varying characteristics and trying to establish the underlying laws of heredity by observing the characteristics of the offspring. So according to this concept, it's discrete particles within cells that determine the characteristics of an organism. Now, through his experiments, Mendel had basically discovered and described the units of heredity that would later on become known as genes. Now, we're not going to get into the actual experiments that Mendel did in great detail here because, as I said before, that kind of level of detail is provided within the course modules on my website. But for the moment, it's enough to say that we can kind of examine generally what he did and the conclusions that he drew and the laws of heredity that he formulated, and then we can see how those relate to our modern understanding of genetics. So, hereditary characteristics are inherited according to certain rules, or what he would refer to as natural laws. Now, excuse me, frog in my throat. Now, the search for natural laws is actually one of the primary aims of science in general, because knowledge of these principles by which natural phenomena occur makes it possible for us as scientists to predict future events, and that's really kind of the fundamental basis of science. It's determining the rules by which natural phenomena work, so that we can then make predictions about how those phenomena will behave under differing circumstances. So, in 1865, he published the results of his experiments on the heredity of garden peas, and it was those investigations that provided the first accurate outline of the mechanisms by which characteristics can be inherited from one generation to another. Now, in fact, his work went unnoticed until about 35 years after his death, but eventually it provided the foundations of the modern science of genetics, because basically what he did, with no knowledge of genes or the kind of structures like chromosomes and alleles and things like that, he essentially established the natural laws which govern how offspring inherit parental characteristics. So he observed that when he was pure breeding parents with contrasting characteristics, so for example breeding a tall plant with a short plant, these characteristic traits don't blend necessarily to produce plants of a mixture of heights. In fact, all of the plants that he produced he found were tall. So he wanted to know what had happened to the short characteristic, which was present in one of the parents but seemingly not present in the offspring. So he conducted further experiments and he discovered the laws which govern these events, and he used them to predict the ratios of certain genotypes and phenotypes in the next generation. And again, this is a fundamental a quality of what we refer to as the scientific method. Now, the scientific method is essentially the way in which experimental science and investigations in science are done, whether you're talking about biology, chemistry, or physics. The essence of it is to find hypotheses which are proven to be accurate by doing experiments to either prove or disprove those hypotheses. If the experiments prove the hypotheses, in other words, if the hypotheses produce predictions which are shown to be accurate by experimentation, then if sufficient quantities of data are accumulated, then those hypotheses become theories, and those theories can be used to accurately predict what will happen under other circumstances. So essentially, he was applying the scientific method to studying heredity in very simple organisms. So he took short plants, he took tall plants, he interbred them, and he found that the offspring were not a mixture of short and tall. In other words, they weren't of an intermediate height. He found that they were all tall, and he wanted to explain this. Now, as I say, we're not going to go into the actual details of his experiments, but it's enough to kind of summarize his results here. So the table here shows a summary of his experiments for 7 distinct inherited traits. So we can see here that he took these characters or these characteristics of plants, and he bred parents with differing characteristics or differing versions of those characteristics, and then he listed the number of the offspring showing the dominant characteristic and the number showing the recessive characteristic, and then he listed those as ratios. So we see here that the ratio of dominant to recessive characteristics usually approximates to around about 3 to 1 for all of these 7 pairs of contrasting traits that we see here. So how did he interpret those results? Well, on the basis of these observations, he came to the following conclusions. So firstly, since the parents of all of the contrasting crosses were purebred, then each variety must have possessed 2 factors for those traits. So 2 tall factors, 2 axial factors, and so on. Now, the first filial generation, which is usually denoted F1, must inherit 1 such factor from each parent for each trait. So these factors are carried by the sex cells or the gametes. Now, he also concluded that the factors don't blend in the first filial generation, but they retain their individuality as discrete particles. And since both those factors are present in the first filial generation, then one must be dominant and the other must be recessive. So when the F1 generation produces gametes, the factors then separate from one another and then randomly combine at fertilization. So essentially, he was making these conclusions on the basis of those observations in order to explain those observations. Now, from these conclusions, Mendel went on to formulate his first law of heredity. Now, this is usually referred to as the law of segregation. So let's have a look at this Mendel's first law here. So, the first law is often known as the law of segregation or the law of segregation of characteristics, and he revealed a predictable pattern of results where the parental generation were pure breeding homozygous individuals of different genotypes. Now, the F1 generation only showed the dominant characteristic. The second filial generation showed the dominant and the recessive characteristics to a ratio of 3 to 1. So the results have been summarized as Mendel's first law, which was stated like this. So in a cross between plants bearing contrasting characteristics, these characteristics segregate or separate in the second filial generation. So this is to say that during a monohybrid cross, the characteristics don't blend together. They retain a distinct individuality and then they separate unchanged in the second filial generation. Now, the second law of heredity that Mendel formulated is sometimes called the law of independent assortment. So, next, Mendel argued that when pollen plants make pollen grains and ova, which are the gametes of the plants, the factors pair and separate so that each gamete receives only 1 factor. So now this is actually explained by what we know about meiosis. So therefore, gametes from a homozygous tall plant have only the dominant factor, which we can label T here, capital T, and the homozygous short plants must only have the recessive factor, which is denoted with a lowercase t here. So, at fertilization, the pollen with the T, the capital T characteristic, and the ova from the tall plants will come together with a recessive pollen and ova from the short plants. So these will produce the heterozygous seeds, like this. So the heterozygous seeds will then grow to produce a first filial generation of tall plants because, again, the dominant factor will always dominate over the recessive factor. Now, he argued that the first filial generation will contain both of those factors, both the dominant and the recessive. So when these plants produce gametes, those pairs of the dominant and recessive characters in the heterozygous individuals will separate, and each pollen grain and each ovule will either receive the dominant or the recessive character. So there are the same number of the dominant and the recessive characters, and therefore an equal number of a capital T and lowercase t ova and pollen grains must be produced. So each type of pollen grain has an equal chance of pollinating each type of ovule. And so 3/4 of the second filial generation will be tall because they have the factors, they are either double dominant, homozygous double dominant, or heterozygous in the opposite format, which basically amounts to the same genotype and will lead to the same phenotype. So the dominant factor will dominate the recessive factor. So 1/4 of the second filial generation will have the factors lowercase t for the double recessive, and so will be a pure line of short plants that you see here. So therefore the results will be again a ratio of 3:1 of tall to short plants. So it's clear from these similarities that the Mendelian factors and chromosomes are linked in some way. So in fact it's now more accurate to say that the chromosomes contain what Mendel referred to as factors, because there are far more factors than there are chromosomes in a cell. Now remember, there are 46 chromosomes in somatic human cells, but obviously there are far more alleles than there are chromosomes. So the term factor is now not used in modern genetics. In fact, now we call what Mendel called factors, we call those alleles. So chromosomes contain many alleles, each of which controls the development of a particular hereditary characteristic. So alleles are usually described as different forms of the same gene that code for different characteristics. So Mendel also stated that in order to achieve the uniform appearance of the first filial generation from pure breeding plants, the 3:1 ratio of the second filial generation, hereditary factors must operate together in pairs. So we now call these allelomorphic pairs. So therefore pure breeding tall plants must have the factors for double dominant factors. So again, we would denote that as capital T, and the pure line short plants must have the factors, must be double recessive for the two recessive factors, lowercase t and lowercase t. So the modern science of genetics is essentially based upon Mendel's observations. He was right, but now we know a little bit more about the actual molecular mechanisms by which all of this operates. So now the modern science of genetics is essentially the study of how genes operate. So Mendel proposed 2 laws of inheritance, and these still hold true in most cases in the modern science of genetics. So let's have a look at how these Mendel's laws apply to what we know of genetics in modern terms. So we need to define some of these terms here. Now, as always, the definitions of scientific key terms are incredibly important, and I would say that they're particularly important for students studying biology because of all 3 of those sciences. Biology is the one that makes the most demands of the student in terms of kind of the rigor of scientific language. So let's have a look at the definitions of some of these important terms here. So firstly, a gene is the functional unit of heredity that carries information from one generation to the next, and it's a specific length of DNA whose nucleotide sequence codes for a specific protein. So we say that a gene occupies a specific position on a chromosome, and this position is referred to as a locus. Now the word locus is singular, the term loci is plural. Now alleles, as we said before, these are alternative forms of the same gene which occupy the same locus on a pair of homologous chromosomes. So alternative forms of a gene will usually arise by spontaneous mutation, and they may alter the way in which a particular characteristic is expressed. So this cell nucleus, as we see here, shows 2 homologous chromosomes, and we see 2 different gene loci in this diagram. So here we see gene locus 1, and here we see the second gene locus here. So we can label these gene loci A and B. Now each gene, as we see here, is represented as 2 alleles. So capital A and small a are alleles of the same gene, dominant and recessive respectively, and similarly capital B and small b are the alleles of another gene, and again these are the 2 alleles, dominant and recessive respectively. So A and B are different genes, but each of those genes has 2 alleles. So, two pairs of homologous chromosomes from the pea plant showing the loci for height and flower position are shown here. So we see here the gene locus for height on these two homologous chromosomes, and we see that in this particular individual, this individual is heterozygous for this gene. And similarly here we see the gene locus for flower position, and again this individual is heterozygous for that characteristic. So here we see the allele for tallness, T being the capital T being the dominant allele. Here is the allele for shortness, which is the recessive allele. Here we see the allele for axial flowers, which is dominant, and here we see the allele for terminal flowers, which is recessive. So the phenotype of this pea plant is tall with axial flowers. Alrighty. Okay, I see that Nell has just logged in. Welcome. Hopefully some of the others will log in shortly, and I'll bring the SWAP Consortium chat up on the screen here. And let me just make myself a little bit smaller on the screen here, just so you guys can see the text a little bit more clearly. Bear with me a second here, guys. Just move myself over a little bit, and we'll make this a bit smaller as well. Alrighty. Okay. So again, let's have a look at those key terms. These are very, very important. So a gene can be defined as a specific segment of DNA whose nucleotide sequence codes for a specific protein. And again, this would make the gene the unit of heredity. The locus is the specific place on a chromosome where a gene is located. Now, the term chromosome is very, very important. And we've defined that in one of the previous webinars where we started talking about cell division by mitosis and also by meiosis. Remember, a chromosome is a structure into which the DNA is organized in order to facilitate the proper distribution of genes to the daughter cells during cell division by either mitosis or meiosis. Now, an allele, as we said before, is an alternative form of the same gene which occupies the same locus on a pair of homologous chromosomes. The genotype is the genetic constitution of an organism comprising all the genes possessed by an individual. And the phenotype is the observable or the measurable trait or characteristic displayed by an organism as a consequence of its genotype. Now, this may be a visible trait, such as flower color, or it could be an experimentally determined trait, such as a blood group or the possession of a specific enzyme. So, homologous chromosomes are pairs of chromosomes of the same size and shape which carry genes for the same traits. Now, one member of a homologous pair is maternal in origin and the other is paternal in origin. The term heterozygous refers to individuals who carry two identical alleles of a gene, and these are described as being either homozygous or homozygotes. So homozygous describes the alleles that the individual has. So in other words, they have two identical alleles, either both dominant or both recessive. Now, an individual who possesses a homozygous genotype for a particular trait is described as a homozygote. Now, heterozygous is used to refer to individuals who carry different alleles for a specific gene on a pair of homologous chromosomes. So again, in this case, the genotype would be dominant and recessive. And these individuals are described as being heterozygous or as being heterozygotes. Now, the dominant allele can be defined as an allele which is phenotypically expressed in both the homozygous or the heterozygous condition. So, for example, the allele for the tall plants, which we gave the symbol capital T in the pea plants, this is expressed in both the homozygous and heterozygous genotypes. Now, the recessive allele, in contrast to that, this is an allele which is phenotypically expressed only in the homozygous condition, where the allele is where the individual has two recessive alleles for that characteristic. So again, the example here would be the allele for shortness, the lowercase t in pea plants, is only expressed when they are homozygous for that allele. Now, you should by now be relatively familiar with the use of Punnett squares, and there are two different ways of drawing Punnett diagrams. So here's one example here, and here's another example here. Now, they're essentially equivalent. They're just two different ways of representing these diagrams. Now, the first of these is called the diamond Punnett, and it does have certain advantages over the box Punnett. And so we're going to be using this format through the course of this webinar here. So, in Mendel's experiment, he engaged with, in doing monohybrid crosses. So if these are the parental phenotypes, here is the male plant and here is the female plant, or rather these are the plants from which the male and female gametes were derived. These are the parental genotypes here, so we see that this plant is homozygous dominant for the tall gene, and this plant is homozygous recessive and therefore has the short phenotype. And so the gamete phenotypes are all dominant in the case of this plant and all recessive in the case of this plant. So the first filial generation is going to be all heterozygous. So this the phenotype for this generation is going to be tall and the genotype is heterozygous. So again, the second filial generation this time, so here we're taking individuals from the first filial generation and crossing them. So those individuals from the first filial generation again were all heterozygotes. So these would be the male gametes in that case, and these would be the female gametes in that case. And so these would be the genotypes of the second filial generation. And so we see here that the 3:1 tall to short phenotypic ratio is expected in the second filial generation. Okay, so we've had a look at some of the monohybrid crosses. Let's now have a look at dihybrid inheritance. So, after completing his monohybrid crosses, Mendel then continued to do more experiments to determine what would happen when two pairs of contrasting characteristics are combined together in the same hybrid. So Mendel chose two different characteristics, such as the height of the pea plants and the colour of the flowers, and then set up crosses to obtain his first filial and second filial generations. So once again, he consistently obtained specific ratios in the second filial generation, and he used these results to formulate the second law of heredity. So here's an example of one of the crosses that he did. He took tall purple flowers and crossed them with short white flowered plants. So the parental genotypes in both cases here, because we're dealing with two characteristics here, we have double dominant for tall and the dominant flower characteristic being purple here, denoted by the capital P. So here this individual is homozygous for both those characteristics and has the dominant alleles, and in contrast this individual is homozygous for the recessive alleles for both of those characteristics. So the gamete phenotypes therefore will all be the dominant alleles for both those characteristics in the case of this parent, and all recessive in the case of this parent. So the first filial generation we would expect to be all of this genotype, the heterozygous genotype for both characters, and therefore with the phenotype tall with purple flowers. Oh, don't know why that happened. Okay, so Mendel allowed the tall purple flowered pea plants to then self-fertilize in order to obtain the second filial generation. So the first filial generation was then crossed with itself, essentially. So to determine the nature of the gametes produced by these F1 plants, we would need to use a Punnett square. And we would need to use that to determine the expected ratio of the phenotypes of the F2 generation. So I'm going to pause here and give you guys a chance to quickly see if you can determine the ratios there. It shouldn't take you very long, but it is a little bit of a tricky problem. So I'll pause and let you guys have a crack at that. So what do you guys think? What will be the expected F2 generation phenotypes? And if you have absolutely no idea, don't worry, because we're going to go through the answers shortly. So I'll give you about a minute or so, and that'll give me a chance to remove the frog in my throat by taking a sip of water. Okay, Okay, so already we have some suggestions. That's actually quite impressive. I'm being a bit evil here because it'll probably take you a lot longer than a minute or so to figure this out, but we've got some bright sparks in both Zoom rooms, so we'll see what you guys can come up with here. Okay. I see that quite a few of you have actually, you're on the right track there actually. Okay. Alrighty, so let's have a look. So here's how we would do this. So the way that we would construct this is we would take the male gametes. Again, it's the F1 generation which is being crossed with itself for two characteristics, because we are studying dihybrid inheritance here. So these would be the male gametes, and these would be the female gametes, and of course these would be the same. So if we cross these, we would expect the following phenotypes and genotypes in the F2 generation. So I guess what I'm trying to kind of illustrate here is that the idea of these Punnett squares, it's equally applicable to monohybrid and to dihybrid inheritance. It's just that you need more squares to cross the available gamete genotypes. So if you're very careful and you kind of understand the way that these alleles separate in the gametes, it's essentially the same procedure that you would follow here. So if we have a look at the genotypes and then count up all of the phenotypes, we find that the expected phenotypic ratio is 9 tall purple flowered to 3 tall white flowered to 3 short purple flowered to 1 short white flowered. So the expected phenotypic ratio for the F2 generation in this case would be 9 to 3 to 3 to 1. So actually quite a few of you got that, which is very impressive. Well done. Okay, so the results of this dihybrid cross show that we expect distinct phenotypes in the F2 generation appearing in predictable proportions. So the expected results, as we said before, would be 9/16 of the progeny would be tall and purple flowered, 3/16 of the progeny would be tall and white flowered, 3/16 of the progeny would be short and purple flowered, and 1/16 of the progeny would be short and white flowered. Now, although Mendel used pea plants because they were very simple organisms with very easy, with characteristics that make them very easy to cross and mature very rapidly. We in modern genetics we tend to use the fruit fly Drosophila melanogaster because it also has a number of features that make it very useful for genetic experiments. So, for example, it completes its life cycle in only 10 days at 25 degrees centigrade. So this allows geneticists to study the inheritance of characters over several generations within a relatively short period of time. And they're also relatively easy to handle in the laboratory, and this allows for many different crosses to be set up for investigation. Another characteristic that makes these organisms quite useful is that very large numbers of progeny are produced with each mating. So this allows for a valid statistical analysis of the results to be of the results that are obtained from these crosses to be analysed. So, male and female adult fruit flies are easily distinguishable, and these allow the sexes to be separated before sexual maturity is reached. So here we can see the characteristics of the female with a pointed striped abdomen, and the males tend to have a rounded abdomen with stripes which merge at the tip. And again, this makes it very easy to separate the sexes, and this ensures that only virgin females are used for the experiments. Now, another interesting characteristic of Drosophila is that they possess 4 pairs of giant chromosomes. So this is one of the giant polytene chromosomes, and so the diploid number for these organisms is 8 chromosomes, and there are many mutant genes which provide geneticists with pools of characteristics for study. So, mutants are inherited departures from the normal or wild phenotype. So, this is the wild type Drosophila, which have red eyes, but white eye colour is actually a quite common mutant character amongst these organisms. So, wild type Drosophila tend to have grey bodies, and sometimes with you see this mutant characteristic of the ebony or black body colour, which sometimes emerges as well. And wild-type Drosophila have normal wings, but one of the common mutations that we see is this vestigial wing, which sometimes occurs as a mutant character. So let's return now to Mendel's second law of independent assortment to kind of explain the results that we saw in that dihybrid cross. Now Mendel actually performed many dihybrid crosses involving two pairs of contrasting characters, and consistently he observed that 9:3 to 3:1 ratio in the second filial generation. Now he argued that unlike pairs of factors, and again he used the word factors, whereas in the modern day we would use the word gene or allele. He argued that unlike pairs of factors in a hybrid could separate from one another into the gametes and then come together again in predictable combinations in the second filial generation. So when two or more pairs of genes segregate from one another at gamete formation, the distribution of any one of them is independent of the other, and this is essentially how we state Mendel's second law, or the law of independent assortment. Now his observations can be explained again in terms of the behavior of chromosomes during meiosis, and again it's important to remember that he himself had no knowledge of the molecular characteristics of cells like chromosomes and genes and things like that, but essentially what he said holds true today for the most part. So let's have a look at this, and again if you want more detail on this, I would encourage you guys to have a look at the webinar that we did earlier in this sequence on the subject of genetics and biodiversity and variation, where we talked about meiosis and the sequence of events that occurs to the chromosomes in meiosis. But let's just quickly have a look at that briefly again in this context here. So if we consider the behavior of the alleles for these two genes on these homologous pairs of chromosomes, so the genotype of this individual would be capital A, small a, capital B, small b, and again we are discussing a dihybrid inheritance here. So during prophase, each chromosome consists of a pair of sister chromatids. The homologous chromosomes will line up in their pairs at metaphase 1, and the arrangement can be shown in either X or Y, like this. And so 4 different types of gametes are possible at the end of the second meiotic division, meiosis 2. So these would be the 4 haploid gametes that are produced by meiosis, starting with this parental nucleus. So 4 different kinds of haploid gametes are produced with equal probability when 2 pairs of alleles, and again those alleles we denoted capital A, capital B, small b, when these segregate during meiosis. So consider the segregation of 3 pairs of alleles this time during the meiosis and determine the number of different kinds of gametes that will arise. So here's another thing for you guys to try. So usually when we're considering problems like this, a tree diagram can be a useful way of determining all the possibilities. So a tree diagram for this particular problem can be constructed like this. So we see that in fact if you work through this problem, and in the interest of time I'm not going to torture you guys with trying to get you to solve this, but I'm pretty sure you guys can figure this out for yourself. It's not particularly difficult. In this particular case, we see that there are 8 different kinds of haploid gametes that can be produced in this circumstance here. So independent segregation, it's due to the fact that 2 or more pairs of alleles are located in different non-homologous pairs of chromosomes which orientate at random on the spindle during metaphase 1 during meiosis. So 2 pairs of alleles segregating independently will give rise to 4 different gamete types. 3 pairs of alleles give rise to 8 different gamete types, and in general terms the number of different combinations possible is 2 to power n, where n is the number of chromosome pairs. So the number of chromosome pairs in man is 23, and therefore the number of possible gametes is 2 to power 23, which comes to 8,388,608 genetically unique gametes. So this is an enormous source of potential variation during sexual reproduction. Now remember that genes located on different pairs of homologous chromosomes will assort independently during meiosis. So genes located on the same chromosome do not assort independently, but are linked together during the separation of the homologous chromosomes during meiosis. So Mendel demonstrated that different types of gametes were produced by dihybrids in equal frequencies by taking the F1 plants and performing test crosses. Now a test cross involves crossing individuals with corresponding recessive homozygote. And this is used to verify the genotype of individuals displaying the dominant phenotype, so for example, double dominant or heterozygous. So in the case where an individual is crossed with a homozygous recessive parent, this is referred to as a back cross. So Mendel observed when he crossed his dihybrids with the corresponding homozygous recessive, and he reasoned that as the double recessive produces only one type of gamete, then there should be 4 different phenotypes resulting from the test cross, and that these should occur in equal proportions. So let's consider a test cross between a tall purple-flowered pea plant and the homozygous recessive. Again, that would be in this case a short white-coloured pea plant. So what we're doing here is a test cross involving 2 individuals with these 2 genotypes for those 2 dihybrid characteristics. So how would we determine the expected phenotypic ratio for this cross? So let's work through this problem here. So here are the test cross parents with these genotypes and phenotypes. If we display the male gametes here, they would look like this, and all the female gametes will look like this. So there's only 1 row necessary for these. So if we cross these, we find the following genotype and phenotype ratios. So we see that a quarter will be the tall purple flowered plants, a quarter will be the tall white flowered plants, a quarter will be the short purple flowered plants, and a quarter will be the short white flowered plants. So Mendel observed this 1 to 1 to 1 to 1 ratio, and this confirmed that the dihybrid produces 4 different kinds of gametes. So in Mendel's pea plants, the homozygous dominants display the same phenotype as the heterozygotes, and a test cross is required to distinguish between them. So these pea plants are both phenotypically tall, but they are genetically different. So in a test cross, the homozygous dominant will only produce 1 kind of gamete, whereas the heterozygote obviously is going to produce 2 different kinds of gametes. So in this case, here are the parent phenotypes, and here are the gamete phenotypes for these test crosses, and here would be the progeny. Now in this case, all the progeny would be tall, displaying the heterozygous genotype, but in this case of this cross, we would find that half would display the tall characteristic and half would display the short characteristic, because of course with this cross we're going to produce half of the progeny with this genotype, heterozygous, and half with the homozygous recessive genotype. So in practice, observed experimental results tend to deviate from the results predicted by drawing out genetic crosses. Now the reason for this, well, there are many reasons for this, but to start with, chance is involved in the production of the different kinds of gametes during meiosis, and in the way that random combinations of male and female gametes come together at fertilization. And so therefore, the sample size of a population of individuals being investigated will obviously have a significant effect on the extent to which the observed and the expected results will deviate from one another. So the sampling error to which the experimental results are subjected to increases when we're dealing with small numbers of progeny. However, the law of large numbers being what it is in statistics, as the sample size increases, observed results tend to approach the expectations more closely. So genetic ratios are analysed using statistical tests such as the chi-squared test in order to determine whether the deviation of observed and expected values is acceptable on the basis of chance. So deviations in results are found not to be acceptable on the basis of chance are said to be significantly different. Now this goes back to some fundamental ideas in statistics, but namely it's this idea of the law of large numbers. So a simple way of demonstrating this is to say that we know that if we do a fair coin toss, then there's a 50% probability that the coin will land heads and a 50% probability that the coin will land tails. Now in any given number of coin tosses, in general, the probability on paper works out such that you would expect that roughly 50% or 50% of those results would be heads and 50% of those results would be tails. However, if you only toss the coin twice, it's reasonably likely that you're going to get 2 heads in a row or 2 tails in a row, even though the expected result just on paper from probability is 50-50. However, if you toss the coin 100,000 times, you're going to find that the results are near enough 50-50, because the larger the sample size, the closer the actual results will approach to the expected results. So let's move on to looking at linkage, gender and sex linkage here. So, the existence of male and female forms within a species is associated with the inheritance of specific sex chromosomes. Now, sex chromosomes are the chromosomes within the entire set of chromosomes of that organism which carry the sex-determining genes. Now, again, humans have 23 pairs of chromosomes in the diploid somatic cells. The 22 pairs of chromosomes that are not sex chromosomes are collectively known as autosomes. So, here shows the entire chromosome complement for a normal human male. We can see here that there are 22 autosomes, and there is 1 pair of sex chromosomes, and in the male this would be of the type XY. Now in a female, we can see here the complement of normal human female's chromosomes. Again, there are 22 pairs of autosomes and 1 pair of sex chromosomes, and in this case they would be of the type XX. So autosomes are all the other chromosomes within the entire set which are not involved in the determination of the sex of the individual. Now there are 22 pairs of autosomes and 1 pair of sex chromosomes within the nucleus of every human diploid cell. Now, sex determination is associated with the inheritance of sex chromosomes, which in most species are referred to as the X and the Y chromosomes. So, let's take a look at sex determination of humans at the chromosomal level. So in most species, including humans, the X chromosome tends to be larger than the Y chromosome, shown here, as they would appear during metaphase. So during gamete formation in males, the X and the Y chromosomes will form a bivalent in prophase 1 and then segregate to opposite poles during anaphase 1. Now, at the end of mitosis, or rather at the end of meiosis, half of the sperms will contain a single X chromosome and the other half of the sperm will contain a single Y chromosome. So the normal human male will possess 1 X and 1 Y chromosome. So this is referred to as the heterogametic sex. Hetero meaning different, gametic meaning gametes. So the male sex is the heterogametic sex because it is the gametic sex which contains the 2 different types of sex chromosomes. Now, normal human females, by contrast, possess 2 X chromosomes, so this would be, the female would be referred to as the homogametic sex. So if we have female and male parents here, these would be the distribution of the sex chromosomes in the gametes. And therefore, if we do a cross here, these would be the progeny. We would see 50% female and 50% male purely by probability. And so we are expecting 50% females with the XX chromosomes, 50% males in the XY chromosome. So the expected ratio in this case is 1:1. So let's have a look at autosomal linkage. So genes that are on the same autosome, not a sex chromosome, are said to exhibit autosomal linkage. So 2 autosomal genes belong to the same linkage group. Now the number of linkage groups is equal to the haploid number, not the diploid number, the haploid number of chromosomes, because each chromosome carries the same gene as its homologous partner. So linked genes tend to show up together in the same combinations in the offspring unless crossing over occurs at prophase 1 of meiosis. So if the linked genes show up together in the same combinations in the offspring at a high frequency, then it turns out that they're close together in the linkage group. So in other words, they're less likely to be separated by crossing over. Now, genes that exhibit autosomal linkage cannot segregate independently during metaphase 1 of meiosis, and the way in which they're inherited is one of the exceptions to Mendel's second law of independent assortment. So, in the classical Mendelian ratios of 3:1 and 9:3 to 3:1, obtained in the F1 and F2 generations from monohybrid and dihybrid crosses, these may be modified as a result of the interaction between different genes. So the classical ratios are seen when single genes control discrete individual characters and when they're located on different chromosomes, so in other words, when the alleles of the genes assort independently. However, when we're considering dihybrid crosses, if the alleles for the two characteristics are present on the same chromosome, then the modified F1 and F2 ratios will be observed, which deviate from these expected Mendelian classical ratios. So this variation is due to the fact that their alleles, again, do not assort independently, and this is what we refer to in genetics as linkage. So the genes located on the sex chromosomes, in other words the sex-linked genes, also give rise to modified ratios. So modified F1 and F2 ratios are also observed when the independently assorting alleles of two or more genes interact in the control of single characteristics, or when there is either incomplete dominance or codominance between the alleles of a single gene. So there's actually a number of characteristics in humans that appear much more frequently in males than in females, and this pattern of inheritance is typical for sex-linked traits, of which there are a number of examples in the human population. Now, the best known example of sex linkage in humans are haemophilia and colour blindness. So let's have a look at haemophilia then. So haemophilia is a condition in which the blood fails to clot normally, and it's a sex-linked condition which is determined by a recessive allele located on the X chromosome. So this diagram here shows crosses between a number of related individuals, and we see the key here to denote their phenotype. So I'm going to leave you with this question here, and we're coming towards the end of the first hour of this webinar here, so I'll let you guys think about this question over the break. So have a look at this diagram and see if you can determine the possible genotypes for individuals in this pedigree. So we can use the symbol XH for the normal allele and X lowercase h for the mutant allele. So, as I say, we've come to the end of the first hour, so again, I will give you guys about a 5-minute break. After the break, we will have a look at how we could solve these problems and how sex linkage occurs in this condition of haemophilia. So again, if you have any questions, I see that quite a few questions have already been posted in the Q&A box for the private Zoom room, and I see Nell is the only person attending from the SWAP consortium. So Nell, until some of your colleagues arise, I'll keep the chat off the screen. But again, have a go at this problem, and I will see you guys in about 5 minutes' time. Okay, everyone, we're back, and I see that nobody has even attempted this problem, so I guess this completely stumped you guys. Okay, well, I tell you what, I will address this problem during the Q&A session at the end of the webinar, so by all means, hold that thought, just in the interest of brevity. Okay, guys, so let's move forward here. So, okay, so haemophilia, as we said before, it's a condition in which the blood fails to clot normally, and famously, it's many of the royal families, particularly the Russian royal family, had haemophiliacs in the family, and the most common form of haemophilia, it's a sex-linked condition which is determined by a recessive allele on the X chromosome. So the inheritance of haemophilia has been documented in the royal family of Queen Victoria, and also Prince Albert and 5 of their children. Now, Queen Victoria and Prince Albert had normal blood, but one of their sons, Leopold, suffered from haemophilia. And as I said before, the Tsar Nicholas II and his wife Alexandria and their 5 children, you can see here in this photograph here. Now, both parents and the 4 daughters all had normal blood, but their son Alexis suffered badly from haemophilia. Now, Alexandria was a granddaughter of Queen Victoria as well. and this is Alexis here, and this was the son that suffered from haemophilia that was famously cured by mystical means, apparently, by the monk Rasputin, who himself is a rather colourful historical character. So, part of the pedigree of haemophilia is in the royal families of Europe. So this would be a diagram illustrating this. So, again, you can use these types of diagrams to determine and identify the female carriers. So, these would be the female carriers here. And this would be a similar way of solving that previous problem. But again, I will address that in the Q&A after the webinar concludes. So let's talk about incomplete dominance, codominance, and multiple alleles next. So when there is complete dominance of one allele over another, the heterozygote phenotype is indistinguishable from the homozygous dominant phenotype. So for example, if you have a pea plant with this genotype, double dominant, tall, homozygous, it will have the tall phenotype. But if you have another individual with who is heterozygous for that characteristic, the phenotype, as long as there is complete dominance, will be indistinguishable from this phenotype here. Now, in some cases, the alleles of a given gene will interact in such a way that complete dominance is lacking. Now, both alleles are expressed in the heterozygote, and the heterozygous phenotype is different from both the homozygous dominant and the homozygous recessive conditions. So this genetic situation is referred to as codominance. Now, sometimes it's referred to as incomplete dominance. Now, it's important to note here that in your reading of various books and various internet kind of revision sources and various other sources for your studies, you might find that some authors and some resources will not make a distinction between codominance and incomplete dominance. However, for the remainder of this webinar, we will make a distinction between these two terms. So I'm going to pause here and see if you guys can give me a definition for incomplete dominance, given what I've just said. So what do you guys think? How would you define incomplete dominance in a sentence? How would you rephrase this information to define incomplete dominance in one sentence? Okay, there's a few suggestions here, and yeah, you're kind of on the right track there. So let's have a look at a rigorous definition for incomplete dominance here. So incomplete dominance we can define like this. So this is the term which is used when neither allele is dominant, and hence both alleles are expressed and contribute equally to the phenotype. So in this case, a heterozygote has an intermediate phenotype somewhere between the dominant and the recessive. And there's a partial influence from both alleles. So let's look at an example of this in the snapdragon family of plants. So in Antirrhinum, the snapdragon, a single gene with two codominant alleles is responsible for the determination of flower colour. So individual plants that are homozygous for flower colour can be either red or white. But the heterozygous plants actually produce a pink flower, as both the red and the white alleles are expressed in the phenotype. So when complete dominance is lacking, the upper and lowercase letters are not usually used to denote the different alleles involved. So in these cases, the gene is represented by a capital letter, and the alleles are given superscripts. So for example, here we could have the symbol C with a superscript R for the allele for red flower colour, and C with a superscript W for the allele for white colour. So therefore, the red flower genotype would be this, the white flower genotype would be this, and the pink flower genotype would be this. So if we take a pure breeding red and a pure breeding white flower and cross them, the gametes from this individual will all be the red color, will all have the red color alleles, whereas the gametes from this individual will all have the white color alleles. So when we cross these, the first filial generation will have this genotype, and because there is incomplete dominance here, both of those alleles will be expressed, and the colors become mixed in the phenotype. So the genotypes and the phenotypic ratio expected in the F2 generation can then be determined in a similar way to the determination of alleles and or genotypes for complete dominance. We use essentially a Punnett square. So here would be how we would determine the F2 generation. So these would be the male gametes, and these would be the female gametes from the F1 generation. When we cross these are the genotypes and the phenotypes that we would expect. So the expected phenotypic ratio is 1 red to 2 pink to 1 white. Now another example of this is the Andalusian chicken. So in domestic fowl, a gene with 2 codominant alleles is responsible for the determination of feather colour. Now in pure breeding Andalusians chickens, the black feather colour is due to the allele which we can designate C superscript B, C for colour, superscript B for black, and the pure white variety therefore can be given the genotype shown by this. So the F1 generation resulting from a cross between the black feathered and the white feathered fowl is made up entirely of slate blue offspring. Now the genotypes and the phenotypic ratio expected in the F2 generation again can be determined in a similar way to before. So essentially we would take the genotypes of the 2 pure breeding parents, separate the gametes, which would be C superscript B in the case of the pure black, C superscript W in the case of the pure white, and obviously under those circumstances we would expect the F1 generation to all be the blue phenotype, which is produced by this genotype here. But then when we're trying to determine the phenotypic ratios in the F2 generation, we are going to be crossing these individuals with each other. And again, to determine that, we can simply draw a Punnett square like this, and we find these genotypes and phenotypes in the F2 generation. So in this case, the expected phenotypic ratio is 1 black, 2 blue, and 1 white. So we've talked about incomplete dominance, but let's go back and look at the term codominance, because again, even though in some kind of revision sources and study resources such as textbooks and online resources, you might find that people are using these terms interchangeably. Now, if we're going to be rigorous in our scientific language, we need to draw a distinction between these. So let's have a look at codominance in contrast to incomplete dominance. Now, codominance can be defined like this. So when both alleles are dominant and are therefore expressed in the phenotype. Now the alleles are independent, so there is no blending of the characteristic. Instead, the phenotype is a result of the full expression of both alleles. So you can see here that there is actually a subtle but very important distinction between codominance and incomplete dominance. So a good example of codominance would be the human MN blood group system. So the MN blood group system in man is an example of codominance which operates in human populations. Now, in addition to the antigens that provide us with the ABO blood group system, there are also 2 other antigens which we designate M and N, which can be found on the surfaces of red blood cells. Now, in the MN blood group system, the groups M, N, and MN are determined by 2 codominant alleles of a single gene. Now the 2 alleles, M and N, are codominant again, and these give rise to the genotypes MM, NN, and MN. So we have group N with this genotype here, we have group N with this genotype here, and we have the blood group MN with this genotype here. So here's a question for you. The inheritance of the MN blood group system is such that there are 6 possible matings within the parent population. So if these are the parents, what would be the progeny? So I'm going to pause here and let you guys see if you guys can figure that out. So what will be the progeny in order from the top of this table to the bottom. So these would be the parents in each case. What would be the progeny? What do you guys think? Okay, so we've got suggestions for all mm for the first row, all nn for the second row. That's fairly obvious. What about the others? All right, seems like you've all kind of got the hang of how to do this. So let's have a look here. So So here's the answer. So firstly, for the first row, if the parents are and crossing, both parents are obviously all the progeny will be so you got that right. If the parents are all NN, the progeny will all be the NN blood group. If the parents are times NN, then all the progeny will be MN. If the parents are times MN, you're going to get half the progeny in blood group half in MN. If the parents are NN crossed with MN, you're going to get half NN and half MN. And finally, if all the parents are MN and crossed with, or both parents are MN, then you're going to get this genotypic ratio in the offspring: a quarter half MN, and a quarter NN. And it looks like most of you got that right. Well done. So now it's important here to mention a couple of things about mutations in the context of codominance. So we're going to look at sickle cell disease to illustrate this. So most mutations, remember, confer no advantage to the organism. The vast majority of spontaneous mutations are actually either silent mutations or they are mutations which confer a disadvantage to the individual. But occasionally, very occasionally, a random mutation may have a beneficial effect. Now, an example of this is sickle cell disease, or SCD. Now, this results from the inherited mutation of the gene that codes for this structure of hemoglobin. Now, the mutated gene which is designated hemoglobin S, this causes the red blood cells to become sickle-shaped. Now, hemoglobin S is recessive to the gene for normal hemoglobin, so only homozygous recessives will suffer from sickle cell anemia. Now, the presence of hemoglobin S genes confers an advantage because it makes these people resistant to a severe form of potentially lethal malaria. So, therefore, the mutated gene, because it does give an advantage, has actually been passed on through the generations, and to the extent that in certain parts of Africa, most of the population actually have this gene and have this disease, because the individuals who didn't have the disease were afflicted by malaria, and this very severe form of malaria killed off many of those individuals before they had the chance to breed, whereas the individuals with the sickle cell condition survived and now make up the majority of the population in many of those areas. So, homozygous recessive individuals are protected from the lethal form of malaria, but often do suffer pain and tiredness as a result of the sickle-celled condition. However, heterozygous individuals are also protected from the malaria. Up to 40% of their hemoglobin may be hemoglobin S. Now, these heterozygotes are said to have the sickle cell trait, but they don't suffer the debilitating effect found in the homozygotes. So expression of both the normal hemoglobin gene and the hemoglobin S gene in heterozygous individuals shows that codominance is occurring in the expression of these genes. So let's have a look at codominance where there are multiple alleles involved. Now again, codominance can be defined as when both alleles are dominant and are therefore expressed in the phenotype. The alleles are independent, and so there is no blending of the characteristics, and instead the phenotype is the result of the full expression of both alleles. So the genetic systems that we've considered so far have been limited to genes with pairs of alleles which control specific characteristics. Now, genes are changed to alternative forms by the process of mutation, and it's possible for a given gene to have more than 2 alleles within a specific population of individuals. Now, whenever there's more than 2 alleles for a particular gene within a population, we have a multiple allelic series. So this is an example of multiple alleles. Now, although there may be more than 2 alleles, excuse me, losing my voice. So, although there may be more than 2 alleles of a gene present within a population, the maximum number that can be possessed by an individual is 2, of course, so with 1 on each of the homologous chromosomes. So a classic example of a multiple allelic series within a human population is the genetic control of the ABO blood group system. Now, the gene controlling the ABO blood group system has 3 different alleles, and each allele codes for the production of a different antigen on the membranes of red blood cells. So the 3 alleles which control the expression of the 4 blood group types that we see here in this diagram are designated I superscript A, I superscript B, I superscript O, and this stands for the antigen A, antigen B, and antigen O. So the allele IA codes for the production of antigen A, allele IB codes for antigen B, allele IO does not code for antigens on the surface of red blood cells. So alleles IA and IB are codominant, and both of these are dominant over the allele IO. So how would we determine the possible genotypes, antigens, and blood groups of phenotypes within this population given this information? Well, we can do it like this. So if these are the possible genotypes, and given what we've already said, then with this genotype, the antigen produced is antigen A, and therefore this individual would belong to blood group phenotype group A. If the genotype is this, remember that the genotypes for both the alleles for A and B are both dominant over the allele O, and therefore again you're going to get the antigen A being expressed on the surfaces of the red blood cells, and this individual would belong to blood group A. If the genotype of the individual is IB, then again we have a homozygote for the allele which produces the antigen B, so it's simply going to produce only the antigen B, so this individual will belong to blood group B. If the individual has this genotype, again, the IB allele is dominant over the IO allele, and so this individual will only produce the B antigen and therefore will belong to blood group B again. With this genotype here, we have both alleles being expressed because they are codominant, and so this individual would belong to blood group AB. And if the individual is homozygous for the IO allele, neither antigen will be expressed on the surface of the blood cells, and so this individual would belong to blood group O. So these blood groups, individuals would only fit into these blood groups if they have these specific genotypes. So have a think about the following problem here. So if the mother is group B and is heterozygous, and the father is group A and heterozygous, determine the possible blood groups for each child born to this couple. So I'll give you guys a moment to consider that, and then we'll look at how we do that. So what do you guys think? See, we've got some suggestions there in the private Zoom room. Nell's been very quiet there on her own in the Acklenburley-Le Swap Zoom room there. So let's see. Okay, so many of you are kind of suggesting the suggestions you're making are going along the right lines. So let's quickly have a look at how we would determine this. So you're only being given a limited amount of information here, but there is sufficient information in this problem for you to work out the answer. So essentially, you would need to, again, produce a Punnett square. So if the parents are heterozygotes with these genotypes, then the heterozygotes can only have these genotypes. So the male gametes would be these, IA and IO. The female gametes are going to be IB and IO. So when we produce our Punnett square, these would be the genotypes and phenotypes of the progeny. So for each individual child, the probability of being of blood group AB, group B, group A or group O in all cases is 1/4. So let's have a quick word about rhesus and ABO blood groups. Now, in addition to classifying individuals as either belonging to the blood group A, blood group B, or blood group AB or O, it's usual to indicate the rhesus status of those individuals. Now, individuals are either rhesus negative or rhesus positive, and this depends upon the presence or absence of an antigen type D on their red blood cells. So rhesus positive individuals possess the antigen type D, and rhesus negative individuals lack that antigen. So a single gene with two alleles is responsible for determining the rhesus state of an individual, whether or not they have that particular antigen type D. So the allele rhesus negative and rhesus positive are the ones that are responsible for that. So Rh plus determines the formation of antigen D and is dominant to the allele Rh minus, which fails to code for that antigen. So the inheritance of the rhesus gene actually follows typical Mendelian patterns for single genes with two alleles. So let's have a look now at the idea of polygenic inheritance, epistasis, and the idea of lethal genes. So in all the examples that we've considered so far, the individual genes and their alleles have been responsible for the expression of discrete single characters such as height or pod colour in pea plants, wing length or body colour in Drosophila, and so on. Now, these single major genes that control single characters will give rise to what we've referred to previously as classical Mendelian ratios. And again, these would be the 3:1 and the 9:3 to 3:1 ratios, which were observed in monohybrid and dihybrid crosses. So this is seen when the alleles of single genes are either dominant or recessive and exert their control over single characters. But as we've seen before in the previous discussion, there are circumstances under which these genetic ratios can be modified. So, the classical Mendelian 3:1 and 9:3 to 3:1 ratios can be produced by crosses that Mendel couldn't have explained. So, for example, phenotypic traits expressed when the alleles of two or more genes interact and exert their control over a single character, so in the examples of polygenic inheritance and epistasis, can lead to these classical ratios, even though if we just follow Mendelian genetics, these ratios would not be predictable under those circumstances. So modified classical Mendelian ratios are also observed in a number of other inheritance patterns. So for example, when incomplete dominance or codominance is involved, there's a lethal genotype and there is a linkage between genes on the same chromosome. So how do we define polygenic inheritance? Well, this term is used when there are multiple alleles of two or more genes interacting with one another to influence a particular phenotypic characteristic. So an example of this in humans would be height, skin color, and eye color. Now, we're not going to explore the polygenic inheritance of those characters in humans. Instead, let's look at something a little bit more simple, which is the form of the comb found in poultry. Now, the comb is the little piece of tissue that you find here, and the shape of that comb actually is a result of polygenic inheritance. So during the course of their work, Bateson and Punnett actually discovered an unusual form of gene interaction which was responsible for determining the type of comb in certain kinds of poultry. So they found that there were 4 different types of comb which exist. So they termed these the walnut, the pea, the rose, and the single comb. Now, when poultry with the pea comb are combined with those bearing the rose comb, the F1 poultry all develop walnut combs. When the walnut fowls are interbred, the second filial generation displays all 4 comb types in that ratio 9:3 to 3:1. But this ratio is produced in a very different way from that of the typical dihybrid cross, because there's only one characteristic involved here. So how does this work? Well, let's explore how this works. So here are the parental phenotypes. So here we have a pure breeding pea-combed fowl, and here we have a pure breeding rose comb. So when we cross these, we find that the F1 generation has a completely different kind of comb, which was termed the walnut comb. Now, if we interbreed the F1 poultry, we find the second filial generation have a distribution of the 4 different kinds of combs to this ratio: 9 walnut to 3 pea to 3 rose to 1 single. So 2 different independently segregating allelomorphic pairs interact to determine the form of the single characteristic comb type. The dominant alleles are P for pea, R for rose, and these are both dominant over the lowercase p and r, the recessive forms, respectively. But there is more to how these alleles interact. So, a genotype with at least one dominant P allele, which can either be this or this, but no dominant rose alleles, has the P comb. Now, a genotype with at least one dominant rose allele, which can be these 2 genotypes here, but no dominant P alleles, has the rose comb. A genotype without any dominant alleles at all has a single comb, and this would be this genotype here. And a genotype with at least one dominant P allele and one dominant rose allele has a walnut comb. So the 2 different dominant alleles interact to give the walnut phenotype. So let's have a look at epistasis now. So we need again to give a rigorous definition for this. So epistasis occurs when there are multiple alleles or of 2 or more genes interacting with one another to control one phenotypic trait, and the presence of one gene suppresses the expression of another gene. So the gene that is suppressed is called the hypostatic gene, and the gene which does the suppressing is called the epistatic gene. So this is the phenomenon of epistasis. So let's look at an example of this in the sweet pea plant. Now, in the early 1900s, Bateson and Punnett crossed white-flowered sweet peas and found that all the progeny turned out to be purple-flowered. Like this. So how do we explain this? Well, when the F1 plants were then crossed together at a specific ratio of purple to white flowers was then consistently found in the F2 generation. So the specific F2 ratio which was observed by these scientists led them to the following conclusions about the flower colour inheritance in the sweet pea. So, firstly, 2 different genes interact with each other to control that single characteristic of flower colour in these plants. One gene modifies the allele of another gene, and again, this matches the definition of epistasis. Now, each gene has 2 alleles, and we can designate these uppercase C and lowercase c for one gene, and uppercase P and lowercase p for the other. So, the gene uppercase C codes for an enzyme which catalyzes the conversion of a colourless compound into a colourless precursor of the purple pigment. Now again, the colourless pigment would appear white, and the gene uppercase P codes for an enzyme that catalyzes the conversion of the colourless precursor into a purple compound. So let's look at another example of epistasis, this time in mice. So fur colour in mice is another one of these examples of the effect of interaction between different genes. Now the wild type mice have what we would refer to as agouti fur, where the individual hairs are black with a yellow band, and this produces the typical brown-grey colour of the agouti fur. The non-agouti mice have black coats composed uniformly of black hairs. Now again, in this case we find that there are 2 different independently inherited genes which are involved in the expression of the phenotype for coat colour. So a single gene with 2 alleles, which we can designate uppercase A and lowercase a, determines the colour of the coat. Now the agouti, homozygous or heterozygous for A, is dominant over the black. The second different gene is required for the hair pigment to be synthesized and therefore expressed. So when at least one dominant allele of this gene is present, the agouti and the black phenotypes can be expressed. When the second gene is in the homozygous form, no pigment is formed and the mice become albino. So let's have a look at the interactions of genes in terms of lethal alleles. So there are quite a few examples within living organisms where a single gene can affect more than one characteristic, and including mortality or death. So normal wild-type mice, again, have the grey-brown fur colour, which is referred to as the agouti fur, but occasionally you find mice with yellow or merle colour fur, which appear in the population. Now, in 1905, the geneticist Lucien Queneau performed crosses with these yellow and agouti mice, and he found that the allele governing the yellow fur is dominant, and that the yellow mice were always heterozygous. So when these heterozygous yellow mice were mated together, their progeny were found to be in the ratio of 2 yellow to 1 agouti, and not the expected 3:1 ratio. So it was eventually found that pure breeding the homozygous yellows were never found, or rather the pure breeding homozygous yellows were never found because the gene for yellow fur is lethal when it occurs in the homozygous condition. Now this was actually confirmed when the dead fetuses were found in the uteri of the yellow females, and they were found to have this homozygous, were found to be homozygous for the yellow gene. Now the Y allele, the capital Y or dominant yellow allele, is dominant in its effects on fur colour, but it's recessive for the lethal characteristic. So again, if we take the first filial generation of heterozygotes and interbreed them, we can expect the following distribution of genotypes and phenotypes in the second filial generation. So the gene, again, for the yellow colour, which we designate capital Y, is lethal in the homozygous form. And so these individuals will basically die before becoming fully developed, whereas these individuals will go on to live and be born. So we find that instead of the expected 3:1 ratio, we find a ratio of 2:1 observed in the progeny for this reason of lethal alleles. So we need to now discuss the next section of the specification. So let me just raise that here. Now, before we go into this, because we've been talking so far about inheritance and genetics, we now need to go into the idea of populations and eventually into determining the frequencies of alleles, genotypes and phenotypes using the Hardy-Weinberg equation. Now, again, I'm not going to go into this in quite as much depth as I do in the course module on my website on statistics. Now, I know that many of you actually already have access to that course module. In this course module, we go through pretty much every statistical test which you need as an advanced level biologist. So if you're interested in the kind of details of the statistics as it applies to advanced level biology, I would encourage you guys to have a look at this course module here. We also go into it in quite a lot of depth in the course module on this topic, genetics, evolution and ecology, on the website as well, but I'm just going to go over it in the kind of minimal detail necessary to cover the spec in this section here. So let's have a look at Hardy-Weinberg, or the chi-squared test first, and then we'll move on to Hardy-Weinberg. So the chi-squared test is the statistical method that allows us to calculate whether a set of observed results differs significantly from the expected results, or whether any deviation from the expected results is simply due to chance. Now, in genetics, the test allows us to compare the goodness of fit of the observed phenotypic ratios with the expected ratios that we determine using things like Punnett squares. Now, the formula used to determine the chi-squared statistic is this, where O is the observed results, E is the expected results, and so remember that this symbol sigma here in statistics means the sum of. So if we summarize some of Mendel's results here from that previous table, again these were the expected phenotypic ratios, which all approximate to around 3:1. Now, when Mendel performed his monohybrid crosses, he concluded that the ratios produced for his 7 pairings of different characters approximated to 3:1. So the tall times the dwarf F1 crosses will be used to determine if the observed results fit a 3:1 ratio. So let's have a look at how we would do that. Now the monohybrid cross produced 1,064 offspring, of which 787 were tall and 277 were dwarf. So the expected number of tall plants would be 3/4 times 1064, which gives 798. The expected number of dwarf plants is 1/4 times 1064, which gives 266. Now the way that we determine these numbers again is that the expected ratio is 3:1, and therefore of the total number of crosses produced, or total number of offspring, we would expect, in theory, 798 of them to be tall and 266 to be short. Now the actual numbers produced were 787 for tall and 277 for dwarf. So the idea here is to perform a statistical test to find out if this deviation in the real results from the expected results is purely due to chance, or whether it's due to some other reason. So, in every statistical test, you start by formulating a null hypothesis, and usually the null hypothesis is of the order where there is no significant difference between the observed and expected results. But the alternative hypothesis, therefore, is that there is a statistically significant difference between the observed and expected results, which is not due to chance. So, here's how we might set out the results. So, we have the table for all of the values that we're going to use to calculate the chi-squared statistic. We enter those values into the table and we perform the calculations indicated here, and then we substitute the values into this and we sum those values, and we find the chi-squared statistic is 0.6065, which approximates to 0.61. So the calculated value here of chi-squared is 0.61, and the number of degrees of freedom is 1, because it's the number of classes, in this case 2, minus 1. So how do we determine whether or not this chi-squared statistic validates or invalidates our null or alternative hypothesis? Well, we need to use a table. So we use standard statistical tables like this one. So the table of chi-squared values like this is used to determine the significance of any observed difference between the observed and the expected results. Now our value of 0.61 lies between 0.46 and 1.07 in the 1 degree of freedom row here. Now, the table tells us for this that the probability value for this occurrence lies somewhere between 0.5 and 0.3. So how do we interpret this probability value? Well, the probability value gives us a measure of the likelihood that any difference between the observed and the expected results is due to chance alone. So in this example, there is somewhere between a 30 and a 50% probability that the difference between the observed and the expected values is due to chance. So when the probability value is above 0.05, which as a percentage is 5%, then again by convention we accept the null hypothesis and we conclude that there is no significant difference between the observed and the expected results. If the probability value is smaller than or equal to 0.05, then we reject the null hypothesis and we accept the alternative hypothesis. In other words, we conclude that there is a significant difference between the observed and the expected results. So in this example, the p-value is above 0.05, and in other words above the 5% probability level, and therefore we can conclude that there is no significant difference between the observed and the expected results. Now again, the observed results we can conclude conform to a ratio of 3:1 because the observed difference is not statistically significant. So let's look at an example of how we can use the chi-squared test to analyse a dihybrid ratio. So purple-flowered sweet pea plants with long pollen grains were crossed with white-flowered plants with round pollen grains. The F1 plants all had purple flowers and long pollen, like this. So the F1 plants, on self-crossing them, gave a second filial generation consisting of the following distribution of phenotypes. So we have 1,528 individuals with this phenotype, 106 individuals with this phenotype, 117 individuals with this phenotype, and 381 individuals with this phenotype in the second filial generation when we take these individuals and self-pollinate them. So we then perform a chi-squared test to determine if these results conform to the expected 9:3 to 3:1 ratio in a statistically meaningful way. So again, we would then organize our results in a table, something like this, just for clarity. We see that the observed ratio is this. So the expected ratio, if we're expecting 9:3 to 3:1, would be this. So again, we then substitute the values into the numerator and the denominator of the equation, and then we evaluate and we sum, and we get a chi-squared value of 966.61 for the 3 degrees of freedom, because we have 4 classes here, and the degrees of freedom is always the number of classes n minus 1. So we have 3 degrees of freedom here. When we relate this to the probability table, we find that the critical value is such that we can reject the null hypothesis. So the probability that the results in this case are due to chance is 3.15 times 10 to the power minus 209. Now this is below the 0.001 probability level, and this is in fact as close to 0 as makes almost no difference. So we can therefore reject the null hypothesis and conclude that the results do not conform to a 9:3 to 3:1 ratio. So let's have a look at population genetics now and the Hardy-Weinberg equation. Now, population genetics is actually the study of the genetic composition of populations rather than individuals. Now remember, the definition of populations in biology and ecology and also genetics is a group of individuals of the same species. So population geneticists study Mendelian populations of sexually reproducing organisms in which there is random mating. Now, a Mendelian population can be considered to be a group of closely related sexually reproducing organisms residing within a defined geographical region. Now, within that region, each member has a statistically equal chance of mating with any other member of the population. Now, obviously this is an assumption, and it's not necessarily true of all human populations, but if the populations are large enough and interact with each other in ways which are complex enough, we can essentially assume that this is the case. So before we get into this, we need to define some very important terms here. So firstly, the term sample. A sample is a representative selection from the parent population. Now the parent population, which is referred to as just the population, is the total group from which that sample is selected. Now the frequency is the total count of a particular variable in a sample, or the estimated count for that variable in that larger parent population. The frequencies are always whole numbers, so for example 105 or 23. Now the relative frequency is the frequency of a particular variable divided by the total of all the variables in the sample, and this is also known as the proportion. So relative frequencies or proportions are always therefore between 0 and 1. Now, geneticists will often use decimals such as p equals 0.5 to represent the relative frequencies of alleles within the populations. So the term frequency is often used instead of the more accurate term relative frequency. So sometimes you might hear somebody say the frequency of allele B is 0.32, but in fact what they should be saying if they're using their language more rigorously is that the relative frequency of allele B is 0.32. Now in this webinar we've chosen the more accurate and more rigorous form of phrasing, where we're going to be saying the relative frequency of allele B is 0.32. So all of this hinges on a genetic concept called or the concept of a gene pool. So for example, we have here a population of 20 fruit flies which was bred in culture bottles in a school laboratory. Some of the flies, as you can see here from this diagram, have normal wings, and these are the long wings which are governed by the normal dominant allele, which we can designate VG+, while the others possess the vestigial wings that you see here, which are governed by the recessive allele VG. So the different genotypes within this population are VG+, so in other words, homozygous for the VG plus allele, heterozygous or homozygous for the VG allele. Now, each individual is described as possessing 2 alleles for wing length, and thus the total number of alleles for this characteristic within the population is going to be 40, which is simply 20 times 2. So the total number of alleles for a particular gene which are present within a population at a particular time, this is defined in genetics as a gene pool. So if we take these 20 fruit flies, so the 20 fruit flies in this population that you're seeing here are all heterozygous for normal long wing length and possess the genotype VG plus VG. Now in this example, there's a total of 40 alleles, again within the population, where 50% of the alleles govern the long wing, in other words the VG plus, and 50% govern the vestigial wing, the VG allele. Now in this example, the relative frequencies of the dominant long wing allele is 0.5 and the relative frequency of the recessive vestigial wing allele is 0.5. So relative frequencies of alleles always provide a total value of 1 when you add them together, where the value of 1 represents the whole population or 100% of the alleles present. So in our example, if the relative frequency of the dominant long wing allele within the population is 0.6, then by definition, the relative frequency of the vestigial wing allele must be 0.4. So there's a general formula which is used to represent the relative frequencies of alleles of a single gene within the population, and the general formula is usually expressed like this, where P is the relative frequency of the dominant allele and Q is the relative frequency of the recessive allele. Now the allele frequencies are essential for analysing the genetics of populations, but they cannot be observed directly. Now, in 1908, the English mathematician G.H. Hardy and the German geneticist W. Weinberg, they developed a mathematical approach in which the allele frequencies, the genotype frequencies, and the phenotype frequencies could be used to study the genetic composition of populations, and this became known as the Hardy-Weinberg equation. So they considered that a population with a hypothetical gene which has 2 alleles, let's just call them capital A and small a, in which capital A is the dominant and small a is the recessive allele, given the frequency of the 2 different alleles, we can calculate the expected frequencies of the progeny genotypes and phenotypes. So here's how we would do it. So if P is the relative frequency of the dominant allele in the population and Q is the relative frequency of the recessive allele in the population, then all chance combinations of gametes containing these alleles can be calculated relatively easily. So it would be similar to constructing a Punnett square. So the progeny would have these alleles with these frequencies, and therefore the expected genotypic relative frequencies in the next generation can be summarized like this. We simply sum those together, and because these must sum to 1, the expression becomes p squared plus 2pq plus q squared equals 1. And this is the Hardy-Weinberg equation. So p squared is the proportion of the next generation expected to be homozygous dominant, in other words, to have this genotype. 2pq in this expression is the proportion of the next generation expected to be heterozygous, and finally q squared is the proportion of the next generation expected to be homozygous recessive. So this is the Hardy-Weinberg equation. So for example, within a population of sheep, white wool is determined by the dominant allele, which we can designate capital B, and black wool is dependent upon the recessive allele, which we can designate lowercase b. So within this population we find that there are 891 white sheep and 9 black sheep. Now we can use this information to estimate the allele frequencies and the frequency and the number of heterozygous white sheep. So how do we do this? Well, so if we let P be the relative frequency of the dominant allele, Q be the relative frequency of the recessive allele, according to Hardy-Weinberg, we can write this expression for these genotypes in the population, and we know that these genotypes in this expression must add up to 1 because they are relative allele frequencies. So all the black sheep are homozygous recessive with the genotype lowercase b. So it's not possible to determine from the phenotype which of the white sheep are homozygous or heterozygous, because homozygous or a heterozygous genotype will result in the white phenotype. So we can calculate the frequency of the black wool allele by first determining the frequency of the homozygous recessive black sheep within the population. So Q squared is the relative frequency of the homozygous recessive alleles, in this case the black homozygous recessive genotype. So therefore Q squared must be 9 over 900, and this simplifies to 1 over 100, which is equal to 0.01. Therefore, if Q squared is equal to 0.01, it means that Q must be equal to the square root of 0.01, which is of course 0.1. So the relative frequency of the recessive allele in this population is 0.1. But since we know that P plus Q is equal to 1, then we know that from this, we know that P must be equal to 0.9. And therefore, the relative frequency of the dominant allele is 0.9. So remember, these two have to add up to 1, so if you know one, you can calculate the other. Okay. So now that we know the relative allele frequencies for the wool colour for these two alleles, we can then again refer back to this expression here. So according to Hardy-Weinberg, the relative frequency of the heterozygotes, again remember, is 2pq. But now that we know what P and Q are, we can calculate that. So the relative frequency of the heterozygous individuals must be 0.18. 2 times 0.9 times 0.1. Now the estimated number of heterozygotes must therefore be 0.18 times the total population, which gives us 162 individuals. Now, a little word about equilibrium conditions. So the Hardy-Weinberg law states that in a large randomly mating population, the allele and genotype frequencies must remain constant from generation to generation. So if a population conforms to the expectations predicted by the law, then the population is said to be in a Hardy-Weinberg equilibrium condition. However, Hardy and Weinberg base their laws on a number of conditions. Now, if any of these conditions are not met, then the allele frequencies and the genotype frequencies will change over time, and this actually is the process of evolution. So the Hardy-Weinberg equilibrium is maintained if all the following conditions are met. So firstly, you need a large population, you need random mating within the population, you need no selective pressures operating, so each genotype has equal fitness and there is no differential mortality between those genotypes. The population must be closed, so in other words there must be no immigration or emigration of individuals from or into another population. There must be no mutation from one allele into another, or if mutation does occur, the forward and back mutations must be equal. And there's no genetic drift, so in other words, changes in allele frequencies resulting from random chance processes, which are most likely to affect small populations. And finally, the final condition is that the characteristic being studied must not be sex-linked. Okay everyone, we seem to have come to the end of the second hour here, so I'm going to give you guys another 5-minute break. After the break we're going to get into the next topic, which is speciation and evolution. Okay, so before we get into discussing evolution, we need to first kind of explore the concept of a species and make sure that everybody kind of understands what a biologist means when they use that term here. So let's have a look at the concept of a species to begin with. So throughout history, biologists have used a variety of different systems for classifying organisms into different groups based upon various characteristics or parameters. But there's rarely been a complete agreement as to what constitutes a species. So biologists consider two principal criteria when classifying organisms as members of the same species. So firstly, members of the same species have to share some common physical or morphological characteristics and display similar biochemical, behavioral, and genetic features that differ from those of other species. Now, members of the same species also need to be able to interbreed and share a common gene pool such that they can produce fertile offspring. So if we want to give ourselves a truly rigorous definition of what constitutes a species, we might want to give a definition something like this. So a species is a group of organisms with similar anatomical and physiological features which can interbreed to produce fertile offspring and remain reproductively isolated from other species. Now, this definition has major limitations when you're considering organisms that reproduce by asexual means, such as prokaryotes, so it's not entirely satisfactory, but for the purposes of advanced level biology, and indeed pretty much any other purpose in biology, it's about as rigorous a definition of the term species as you're likely to get. Now, despite the acceptance of this definition by the biological community, it does not provide us with a universal definition which is applicable to all groups and presents some difficulties when tracing the evolution of species from the fossil record. Now, obviously, dead organisms can't be tested for interbreeding capability, so the definition cannot be applied to self-fertilizing organisms or to those that rely almost entirely on asexual means of reproduction. Now, asexually reproducing organisms are usually classified as members of the same species when they share similar morphological, physiological, and biochemical features. So in sexually reproducing populations, the ability to interbreed in their natural environments and to produce fertile offspring is considered to be the ultimate test in determining whether or not organisms belong to the same species. So how do different species arise? So the term speciation, or the origin of species, refers to the formation of new species from existing ones. Now, through interbreeding, the flow of genes, or gene flow, occurs between populations of a species, but not between populations of different species. So for populations to become different species, there must be some kind of barrier which prevents gene flow between them, so that they can become reproductively isolated and diverge. Now, a barrier that prevents gene flow between populations is referred to as an isolating mechanism, and there are several such mechanisms which have been described by means of which a new species may evolve from an existing species. So, we can talk about either geographical isolation or reproductive isolation. So, reproductive isolation, we can then go on to talk about post-zygotic or post-mating barriers such as hybrid fitness. In other words, the hybrid is fertile but weak and often has developmental abnormalities. Or the hybrid may be sterile. In other words, the hybrid survives but is sterile and unable to reproduce. Now, reproductive isolation can also be spoken about in terms of prezygotic or pre-mating barriers, and these can be ecological, they can be temporal, they can be behavioral, or they can be in terms of incompatibility. Now, in the ecological context, prezygotic barriers involve populations living in the same region but occupying different habitats. With temporal barriers, populations reproduce during different seasons or at different times of day, and this is what prevents them from mating. There may be behavioral barriers where animal populations may become isolated by changes in their courtship behaviors, or successful mating may be prevented by the development of anatomical or physiological differences between those populations. So the two principal models that have been put forward to describe the effects of isolation mechanisms in the process of speciation are firstly allopatric speciation, which comes from the word allo meaning different and patric meaning country. So in this case, geographical isolation is required before the two populations can diverge and become separate species. Now again, geographical isolation prevents gene flow between the isolated populations, which can then become reproductively isolated. But you can also look at this in terms of the model of sympatric speciation, and this comes from the term sim meaning the same, and again patric meaning country. So sympatric speciation tends to be less frequent than allopatric speciation, and this arises when an isolating mechanism disrupts the gene flow within a population that resides in the same geographical region, and this results in the creation of two or more reproductively isolated groups. So, if we imagine an original population inhabiting a forest ecosystem and sharing a common gene pool of species A, so there might be a geographical barrier such as a rising sea level that then isolates that population into three subpopulations. So we can call those populations C, A and B. Now the separate islands can then become different environments as climatic and other factors can influence the physical natures of those environments. So now, natural selection in these three different environments will act in different ways on the variable populations, and this promotes differences in their genetic compositions, and this in turn enables them to adapt independently to each environment. So eventually, the consequence of this is that these subpopulations will diverge to such an extent that they become reproductively isolated from one another and therefore become different species. So this would be a model of the mechanism or one model of the mechanism for allopatric speciation. So let's look at geographical isolation in a bit more detail. Now the classic example of this in biology was presented by Darwin with his finches. So in 1835, towards the end of a 5-year voyage, Charles Darwin arrived at the Galapagos Islands. And these are a group of 13 large islands and many small ones spread across the equator in the Pacific Ocean. And these are situated around about 600 miles west of the coast of Ecuador, and Ecuador actually holds sovereignty over these islands. Now, these have never formed part of the mainland of South America, and we can confirm this from geological studies. Now, various species of animals and plants came across the ocean to inhabit these islands, where they encountered very little competition, and thus became what we would refer to as founder species. Now, Darwin was particularly interested in the Galapagos finches, which he later believed to be descended from a type of mainland finch which invaded those islands. Now, 14 different species of finch now inhabit the various islands of the Galapagos, and these vary considerably in their physical characteristics, in particular in their beak shapes and also in some of their behaviours, like their feeding habits. So, Darwin visited 4 islands, and he spent a total of about 5 weeks observing, collecting, making notes, making diagrams, and the islands visited were Chatham, Charles, Albemarle, and James Islands. So he made a special study of the 14 different species of finches found on the islands, and he noticed that each island had its own particular combination of species, and some of these species were unique to just one island. So he suggested that one ancestral species of finch had arrived from the mainland and eventually spread to all of the islands. So this is geographical isolation, and the island's different environments caused the evolution of these different species. Now he proposed that as the populations of finches inhabiting the various islands increased, then the competition for food would also increase. Now natural selection acting upon the variation within each finch population resulted in the evolution of various beak types, and the finches then became adapted to fill the available food niches. Now, these divergent populations eventually developed into different species as other variations, such as mating behaviours, became barriers to interbreeding. Now, many of the finches continue to live on separate islands as allopatric species, whereas now others live as sympatric species on the same island. So let's take a look at some other examples of reproductive isolation. So, seasonal reproductive isolation, as we said before, occurs when populations exist within the same ecological area but are sexually mature or sexually active at different times of year. So this is seen in various American frog species. So we see here that the wood frog becomes particularly reproductively active in April, whereas the tree frog sees its height of reproductive activity in June, and the bullfrog in July. So different species ensure successful mating through the evolution of species-specific courtship behaviours. So a good example of this would be the male peacock. So the peahens are stimulated to mate only when they encounter the specific courtship display of the peacock. The larger the male's feathers and the more attractive the patterns are to the female, the more likely he is to attract a female and successfully mate. Now, there are quite elaborate courtship behaviours displayed by many other species of birds, such as the mallard duck, and these ensure that the males normally only respond to their own species. Now, the feathery antennae of various other insect species possess specialized cells for detecting the sex attractants, or the pheromones, released by the females of the same species. Now, these pheromones are very, very species specific. Now, a number of plant species exhibit mechanisms to ensure that fertilization only occurs between members of their own species. So, for example, in certain clover and grass species that you see here on the left and on the right respectively, the stigma secrete proteins that prevent foreign pollen grains from germinating. Now, in sympatric speciation, the species diverge without geographic isolation or changes in habitat. Now, as we said before, this is comparatively rare, since even a small amount of gene flow may remove genetic differences between parts of a population. Now, generally, sympatric speciation in animals requires the evolution of genetic differences and also non-random mating to allow the reproductive isolation to evolve. Now, disruptive selection is one of the main forces that drive sympatric speciation in natural populations. Now, the pathways which lead from this disruptive selection to sympatric speciation, these are not often prone to deviation. The pathway is actually a kind of domino effect, which depends upon the consistency of each factor. So, these pathways often consist of disruptive selection, being based on intraspecific competition, in other words, competition between members of the same species, which may lead to reproductive isolation and finally to sympatric speciation. Now, when this kind of intraspecific competition is not at work, disruptive selection can still lead to sympatric speciation, and it does this through maintaining polymorphisms. Now, in again the course module dealing with this subject on my website, we do go into this in far more detail, and we also explore the concepts of polyploidy and the implications to speciation, but because this doesn't appear in the AQA spec, I'm going to omit that from the discussion here, just for the sake of brevity here, so that we can cover as much as we can in the remaining time of this webinar. So let's have a look now at different kinds of natural selection. So, natural selection can affect allele frequencies within populations in several different ways. Now, there are three types of selection which operate within populations, and these are stabilizing selection, directional selection, and disruptive selection. Now, for continually varying characteristics, these selection methods will modify the frequency distribution in various ways. So, disruptive selection, which is also sometimes referred to as diversifying selection, this is relatively rare and particularly rare in animals. Now, this is a form of selection which favours the extreme phenotypes of a particular trait over the intermediate forms, and it's a selective pressure that selects against the original population mean and against the intermediate forms. So these tend to be the heterozygotes. Now, the end result of this is that the population affected becomes divided into two very distinct groups, and this would be referred to as a bimodal population. So, when the disruptive selective pressure is exerted, the mode is disfavored, whereas the extremes of the phenotypic range are favored, such that the frequency distribution of the phenotypic characteristics results in a bimodal population. So let's have a look at an example of this. So the organism that you see here is Papilio macaon, which is the swallow-tailed butterfly. So this can be seen in this European butterfly. Some larvae will feed and pupate on plant leaves or stems which are brown, whereas others will feed and pupate on plants with green stems or leaves. Now, over time, two distinct coloured forms of the larvae are seen, and these are brown and green. But very few intermediates are found because these are at a selective disadvantage because they are more present or more visible to predators on the two types of plants. So, disruptive selection is one of the main forces that drives sympatric speciation in natural populations. Now again, sympatric speciation arises when an isolating mechanism disrupts the gene flow within a population that resides in the same geographic area, and this results in the creation of two or more reproductively isolated groups. Now again, the pathways which lead from disruptive selection to sympatric speciation are not often prone to deviation, and it's a domino effect which depends upon the consistency of each factor. So these pathways often consist of disruptive selection being based on intraspecific competition, and this leads to reproductive isolation and eventually to sympatric speciation. So when the intraspecific competition is not at work, disruptive selection again can still lead to sympatric speciation, but it does this through maintaining polymorphisms. So the next type of selection that we're going to look at is stabilizing selection. Now, stabilizing selection does the opposite. It acts against the extremes within a range of phenotypic variation in a population. It actually favours, or rather the type of selection leads to a reduction in the range of variation within the population, but does not change the mode. So stabilizing selection tends to operate in an unchanging environment to maintain the best adapted genotypes within the population. So stabilizing selection, for example, occurs in the selection of birth mass in humans. Infant mortality is greatest for babies of either very high or very low birth masses. So we see here that because of this, we see that this is an example of stabilizing selection because the mode for that characteristic is being favored. Now finally, directional selection. This operates in changing environments, and it acts either for or against the extremes of the phenotype within that variable population. Now this is the main type of selection practiced by man when they artificially select domesticated plants and animals for the improvement of stocks. So this type of selection will lead to a reduction in the variation in the population together with a progressive shift in the mode. So an example of this would be in giraffes. So the evolution of the long-necked giraffe is also thought to have arisen by directional selection. So the ancestral giraffes would have displayed variation in neck lengths with random spontaneous mutations giving rise to giraffes with a slightly longer neck than the average of the population. So during periods where food was scarce, longer-necked giraffes would survive because they'd be able to reach the available food which other giraffes couldn't reach. So natural directional selection would favour the long-necked giraffes, and these variants would therefore survive and be more likely to breed and pass their genes on to the next generation. So in other words, long-necked giraffes were at a selective advantage during periods of food shortage. Now again, we don't have time to go into many other examples of directional selection. For example, we could talk about copper-tolerant grasses, sickle cell anemia and malaria, industrial melanism, the land snail, and so on. But for the sake of brevity, we'll just stick to this one example here. But there are many, many other examples. So selection pressures operating against the shorter-necked giraffes when food was scarce. This means that eventually the frequency of the, distribution of the phenotypes in that population would show a shift in the mode towards the longer lengths of neck. So in time, the longer necked giraffes would become more frequent within the population. Now, one other example that I will bring up of directional selection is the evolution of antibiotic-resistant bacteria by directional selection. So you start with a bacterial population, you have a high rate of spontaneous mutation in such populations, which can lead to individuals of that population becoming resistant to specific antibiotics. Then, when that population is exposed to the antibiotic, the susceptible individuals are all killed off, and this leaves only the mutant individuals which are resistant to that antibiotic, which will then multiply rapidly and form the bulk of the next generation. So let's move into the ecology segment of this section of the specification of this topic. Now, I will say that in this section, there's quite a bit of practical techniques which are referred to here, such as belt transects, quadrats, but of course there are other practical techniques such as Tillgren funnels and kick sampling and various other things. Now, I know that all of the attendees in the private Zoom room, and also Nell, who's in the LeSwap consortium Zoom room, I know you guys all have access to both this course module and also the course module on practical techniques in biology, so you will already be aware of these practical techniques. So I'm not going to, in the interest of saving time, I'm not going to refer to those techniques in too much detail. I'm simply going to explore the actual ecological content of this topic. But if you are interested in more of that, you'll see that, for example, this course module on this topic also includes a number of lessons on ecological techniques, including the application of those ecological techniques to various types of environments in field work, but also it contains quite a bit of teaching on the statistical applications involved in those types of field work as well. So if you're interested in that, by all means explore this course module and also the course module on statistics, which goes into the various statistical tests in more detail, and also provides you with a number of pre-configured spreadsheets to allow you to understand those statistical tests in more detail. But also this course module also contains some PDF guides to those different types of statistical tests, which you can also download as well. So I would encourage you guys to do that, because frankly we are running out of time and we've still got quite a bit to cover in this webinar as well. So let's get into the ecology section now. So we're going to need to refer to, we're going to need to make sure that we are confident in being able to define some very key ecological terms. So let's start by looking at the very general ecological term, the term biosphere. So living organisms occupy a relatively narrow zone of air, water and land, which we refer to as the biosphere. Now the biosphere represents only a very thin layer in relation to the size of the Earth. So ecology is the scientific study of the interactions between organisms and between organisms and their environment that determine the distribution and the abundance of those organisms within the biosphere. Now, ecologists do employ a variety of techniques to investigate, firstly, the relationship between a single species and its environment, the relationships that exist between individuals of different species within a community and also between them and their environment, the changes that take place in the abundance, distribution and activity of a species. So ecology is actually a multidisciplinary science. It encompasses techniques and concepts from physiology, chemistry, mathematics, physics, biogeography and many other disciplines. So biomes are the world's major communities which are classified according to the predominant vegetation and are characterized by the adaptations of organisms to that particular environment. Now each biome consists of many ecosystems whose communities are adapted to the prevailing environmental conditions. So the biomes are classified in various ways, but they're generally grouped into types that describe deserts, forests, grasslands and tundra. So different global sea temperatures are indicated by this thermal image of the Earth. So sea temperatures are actually a major factor in structuring our climate and therefore in defining the boundaries of the different biomes. Now the climate and the geography of a region will also determine the type of biome that can exist in that region. So temperature, rainfall, light intensity, these are the major environmental factors that determine the nature of a biome. So high temperatures, abundant rainfall and roughly constant day lengths throughout the year, these are the conditions that produce the tropical rainforests, for example. So this map here shows the major biomes of the world here, and again in the course module we go into examples of each one of those biomes in a lot more depth. So what exactly is an ecosystem? So I will pause there and let you guys answer that question. How would you define the term ecosystem? I'm rapidly losing my voice, so hopefully this little sip of my tea should fix that. So, how would you guys define the term ecosystem? Ooh, there's quite a diversity of suggestions here. All right, so be careful with these terms, everyone. This is there are so many specific key terms in biology that, and so many marks available in exams which involve kind of descriptive, explanatory, and extended written response questions that you really need to take the time to memorize. And I do mean memorize the formal definitions of these terms. Now, I know that's time consuming, I know it's difficult and nobody likes to do it, but if you really want to get the top marks, you need to be very confident in being able to define these terms for two reasons. Firstly, there's a significant number of marks at advanced level which are available simply for giving the definitions of key terms. So you're going to see a lot of questions which are worth 1, 2, or maybe 3 marks for defining a particular key term. But more importantly than that, you're going to see an awful lot of questions and also essay questions worth 25 marks, which require you to make use of those key terms in a rigorous and appropriate manner. And you're not going to be able to do that unless you can define them. So memorize these definitions, guys. So let's start with how we define an ecosystem. So first of all, we need to be aware of the fact that living organisms are constantly interacting with each other and also with their physical environment. So, living organisms and their physical environments function as an organized unit called an ecosystem. So each ecosystem is a functional unit in which energy and nutrients flow between the physical, non-living, abiotic environment and the community of living organisms that make up the biotic environment. So, for example, the productivity of agricultural plants is affected by a variety of abiotic factors. So these would include things like environmental temperature, light intensity, carbon dioxide concentration, availability of nutrients, and many others. Now, commercial plant growers will manipulate these abiotic factors to provide a greater yield and greater profit. So an understanding of ecosystems is essential to applied biology, whether it's in agriculture or in other branches of applied biology. So an ecosystem is a natural functional unit which consists of several habitats and their communities. So it thus consists of both abiotic and biotic factors and encompasses their interaction. So the habitat of an organism is its place of residence. This is where it lives. And so, for example, things like the bottom of a lake, in a cave, within a tree hole, these are all habitats for particular organisms. Now, to remind you, a population is defined as a group of individuals within the same species which occupy a particular habitat. A community, on the other hand, is a collection of different populations which occur together in the same space and at the same time. So the environment of organisms is the set of conditions that surrounds them and encompasses both their non-living or abiotic and their living or biotic environments. So please make sure you memorize these definitions and that you're able to recount them from memory so that you end up using them appropriately in your written responses. So all animals and plants within the biosphere have to contend with certain pressures from the non-living and from the living environments in order to survive. So abiotic factors that can affect an organism's survival can be things again like light intensity, temperature, water availability, availability of inorganic ions which are particularly important for plants, the availability of space or territory, carbon dioxide or oxygen concentrations, and many others. Now, the biotic factors, or the factors from the living environment which can affect an organism's survival, can be things like the availability of food, and this includes predator-prey relationships, the availability of a mate, the occurrence of parasitism or disease, and so on, and there are many others. So there's a very important concept in ecology which is central to understanding ecology in general, and that is the concept of the ecological niche. Now, the ecological niche of a population is its role within the community, and it's described as it's defined as the sum total of all aspects of the population's existence, and this includes its habitat, its activities, its requirements together with its effects on both the abiotic and the biotic environment. So the important thing to understand here is not just how we define the ecological niche, but the importance of the concept of the ecological niche arises because the ecological niche is the outcome of evolution. A species will acquire a range of adaptations through natural selection, and these will establish for that species the range and boundaries of the ecological niche. So every organism of a particular species is adapted to survive within a range of environmental conditions. Now, this range of environmental conditions within which organisms survive are described as the tolerance ranges, and these include those factors that we referred to before: temperature, light intensity, rainfall, and so on. So, in addition to intraspecific and interspecific competition, in other words, competition between members of the same species and competition between members of different species respectively, there's a wide variety of different types of interactions which occur between organisms within the natural world. And again, these include predation, parasitism, symbiosis, and so on. So predation refers to the interactions between different species where one organism, the predator, obtains its food by killing and eating another organism, the prey. Now, studies of predator-prey relationships have shown that these populations are regulated by negative feedback mechanisms which maintain those populations at a level that can be supported by the environment. So when predator-prey populations' densities are monitored over a period of time, we find that there are cyclic oscillations in their numbers. So, for example, predation by lions is one of the factors that keeps the population of zebra in check. The populations stay strong because the weak and the injured are the ones which tend to be killed by these predators. So, if we track the population numbers of both the predator animals and the prey animals over time, we find that an increase in the population density of the prey results in a corresponding increase in the predator population. And also, when the prey population decreases, this results in a corresponding decrease of the predator population. And so we see these cyclic oscillations in over the course of time. Now, typically we find that the predator curve lags behind that of the prey, and this makes sense because as the prey population declines, there is increased competition between predators for the remaining prey, and eventually the predator numbers begin to fall. But when the predator population declines, the prey population starts to build up again because fewer of them are being killed by the predators, and this again is followed by an increase in the predator population because there's more food for them. So here we see a predator and one of its prey animals. So this is a classic example of the predator-prey relationship, is that of the relationship between the Canadian lynx and the snowshoe hare. Now, between 1845 and 1930, the number of lynx and hare furs sold by trappers to the Hudson Bay Company in Canada were recorded and then graphed. And this was the graph that was produced. So we see here that increases and decreases in the number of hares triggered increases and decreases in the number of lynx and vice versa. So factors other than predation are also likely to have affected the predator-prey populations. So things like, for example, the availability of plants for the hares and the effects of disease and climate changes on the lynx. And these are likely to have contributed to any fluctuations within these populations. But again, in general, we see that the curves follow each other, with the predator curve in general lagging behind the prey curve. So if we remove the dominant predator from a particular food web or food chain, so for example, in this case the starfish, removal of that starfish allows the barnacle population to grow dramatically. Chitons, limpets, anemones and sponges will then compete with the barnacles for food and space. These organisms are then outcompeted by the barnacles. So in nature, starfish control populations of their prey by keeping their own numbers relatively low. So starfish preferentially feed upon the most abundant prey, such that any increase in either of their prey populations would be kept in check by this selective feeding behavior. So in this example, the chiton and the limpet populations are maintained within the community when their predator is present, but face elimination when the predator is absent. Now, as I said before, I'm not going to spend too much time on ecological techniques, so I'm going to quickly skip past here. But just to remind you guys, these are the we're referring here to the kind of practical methods that ecologists use for investigating both the biotic and the abiotic components of a particular ecosystem or environment. So the key to all of these methods is the fact that you ultimately have to be aware that however many organisms you are sampling or measuring the numbers of, that represents a small sample of the total population, and therefore you are reliant upon statistical techniques for analysing that data. So at the core of all of those statistical techniques is the necessity for random sampling. Now, random sampling is necessary because it eliminates bias, it reduces the effects of chance, and it minimises errors in obtaining measurements. So the most common techniques are things like quadrat and transect sampling, but there are also many other techniques. And again, if you want more details for that, I know that all of you currently in attendance already have access to those two course modules, both on ecology and on statistics. So I'd invite you guys to have a look at the resources on my website in those course modules. So I'm going to skip past things like the Tillgren funnel, the so-called PUTA, kick sampling, and I'm going to skip past these bits on quadrats because I know you guys already have access to some in-depth teaching videos on this subject. So we've got about 20 minutes left, and I'd say probably about 3/4 of an hour left in terms of the stuff I need to cover. So I'm going to skip past all this stuff on ecological techniques, and I'm going to go right the way through to the idea of succession, because I know that many of you wanted me to cover that. So unless you guys have any objections, please do let me know if you want me to go back and explore any of these things that I'm skipping over. I'm just trying to make sure that we get to the term succession. And of course, if you'd like me to skip back, or if you have any specific questions about any of those ecological techniques, please do type those in the Q&A box, and I will address those at the end of this webinar. So let's talk about succession then. So ecosystems are dynamic units. They are subject to constant change. Now, this includes both the biotic and the abiotic components of the ecosystem. So in newly formed habitats and in areas subjected to a disturbance such as a fire or a flood, changes in the biotic community follow in a process that we call succession. So if we want to give ourselves a rigorous definition of ecological succession, we could do something like this, or we could give something like this. So ecological succession is a directional change in the composition of species that occupy a given area through time, and it involves initial colonization and the establishment of pioneer plant species, followed by their replacement with other species until a relatively stable community has formed. So communities that tend to remain stable over long periods of time are referred to as climax communities. So here's one such community, an example of one such community, a mature woodland or forests, and also grasslands. Now, at each stage or sear during the progress of succession, communities will modify their environments and create conditions that favour the growth of other species. So ecologists will recognise two types of succession: primary succession and secondary succession. Now, primary succession occurs in newly formed areas where no community of living organisms has existed before. So, for example, sand dunes, new volcanic islands, ponds and lakes and bare rocks. Secondary succession occurs when there's an established habitat which has been subjected to a disturbance, and where some soil and living organisms remain in that habitat after the disturbance. So when we refer to disturbances here, these can be things like natural disasters such as fires and floods, but they can also be the effects of human practices such as clearing forests for monoculture or lumber production and so on. Now, secondary succession can also occur on abandoned agricultural fields. So let's have a look at the events involved during primary succession. So we start with an empty habitat, a harsh habitat. That habitat is colonized by initially pioneer plant species. Now, I would argue that this is somewhat of a simplification, because if you want to be even more rigorous about it, usually before the colonization by pioneer plant species, these habitats can be colonized by fungal species, which can then lay the appropriate groundwork, as it were, for the colonization by plants. But anyway, once the habitat is colonized by pioneer plant species, those pioneer species will modify the environment, which will then create conditions necessary to support new species. So the establishment of new plant species increases the species diversity as various groups of animals will enter the habitat. So the early colonizers are then replaced by new species, and eventually you will get the development of a climax community. So let's look at an example of this. So if we imagine that we have an environment of bare rock. So bare rock is an incredibly inhospitable environment for most organisms. There's a lack of soil to establish rooting structures, together with a lack of nutrients and water for supporting higher plants. So once again, abiotic factors dominate here in determining which species can colonize this kind of harsh environment at the very beginning of primary succession. So typically you will find that lichens are a typical pioneer species which can colonize the bare rock, as their structure enables them to penetrate even the smallest of cracks and fissures in that rock. So lichens are composed of a fungus and algal cells that are associated together in a mutualistic relationship. So the fine hyphae, those filaments that the fungi produce, start by penetrating the tiny fissures in the rock, and they secrete acids such as oxalic acid, which can start to break down the rock. Now the algal cells can then photosynthesize and supply the fungus with organic food substances. So here we can see a lichen slowly eroding this rock. So the combination of the chemical erosion by acids such as oxalic acid produced by the fungal mycelium and the weathering will gradually crumble the rock, and this is what initiates the formation of soils. Now when the lichens die, they add humus or organic matter to the soil, and of course this makes the soil more fertile for more plant species. So eventually you'll find a thin layer of soil and organic matter accumulating, and this allows simple plants such as moss spores and grass seeds to germinate and grow. Now these organisms then continue the process of breaking down the rock and slowly contribute to the organic matter of the building up of the soil when they die. So eventually the improved soil quality is able to support the growth of higher plants, which then outcompete and gradually replace the pioneer species. So now you have a new community of grasses, mosses, ferns and flowering plants, which eventually replace the lichens. So in favourable climatic conditions, succession continues, and the enriched soil may eventually support the growth of shrubs and trees. So biotic factors at this point become increasingly important in determining which species colonizes the habitat as the succession proceeds from there. So to summarize, we start with bare rock. You then go to a lichen-dominated community on the bare rock, then the mosses and the ferns, then the grasses and the flowering plants, and then eventually a climax woodland, given enough time. So another example of primary succession that you may well have come across already is that on sand dunes. Now sand dune succession begins with the formation of mobile embryonic dunes, where the pioneer grasses stabilize the more complex or the mobile sands and prepare the way for the establishment of more complex communities. So the pioneer species are plants that are able to withstand the harsh conditions of the early unoccupied dunes. So this would start with things like sea spurge and holly. So these organisms can colonize bare sand, and they're tolerant to high salt concentrations. So typically in these environments, these form the typical pioneer species. Now these, as I say, these types of species are tolerant of high salt concentrations, but they're relatively intolerant of being buried by sand. So pioneering grasses are then responsible for the formation of embryonic dunes. So the sea holly and sea spurge are the pioneer species found colonizing the bare sand close to the strand line. Now these are intolerant of being buried by sand, so the pioneer grasses are responsible for the embryonic dunes development. And then sand couchgrass is a pioneering grass with an underground stem or rhizome, which is able to grow to great depths. So the embryonic dunes will form as the sand collects around these couch grasses, and then the rainwater causes the salt to leach away from the sand. Now at this stage, the dunes are unstable as wind-blown sand collects at their surfaces. The sand is dry and the humus content of organic material is almost non-existent. So the early dunes are also subject to extremes of temperature and harsh windy conditions, and these again in turn promote water loss and potential desiccation of those colonizing species. Now, under the influence of rainfall, as the salt content of the sand diminishes, you then get marram grass, which then colonizes and dominates the dunes, and this is responsible for the formation of the more stable yellow dunes. So, marram grass is a xerophyte which possesses adaptations for conserving water, and these include the structural features for reducing water loss and for obtaining limited supplies from great depths in the sand. So, this is a section through one of the enrolled leaves, and you can see here that the stomata occur at the base of these deep folds in the leaf, and this is one of the adaptations of these kinds of xerophytic plants to prevent excessive desiccation. Now, the vertically growing underground stem, or the rhizomes of these plants, as I said before, these can extend to great depths in the sand, and this is one of the things which helps stabilize those sand dunes. Now, adventitious roots will then grow laterally and extensively from that rhizome, and this enables the grass to obtain sufficient water despite the fact that that environment may well be quite dry. So here we can see some of the structural adaptations, and these ensure that a humid atmosphere is kept inside this enrolled leaf, and this prevents, as I say, excessive desiccation. Now if we zoom in on one of these pits here, we can actually see that the hinge cells here enable this leaf to roll inwards, and you can see here the stomata near the bases of these deep folds, and in turn that prevents excessive desiccation by maintaining a humid atmosphere inside the leaf. The other adaptation is that the leaves have these hairs, and again these hairs are designed to trap that humidity. So the vertical size of the rhizomes is dependent upon the depth of sand cover. Now the more sand that's deposited, the more vigorously the plant will grow. Now marram grass is the dominant plant of the yellow dunes, where it stabilizes the sand and allows these dunes to grow up to heights of 30 meters. Now the height of the marram grass in turn will alter the microclimate as the wind strengths are broken and less sand gets blown onto the yellow dunes. Now as this microclimate changes and the dune stability increases, the tussocks of marram grass begin to break up, and these are broken down by soil bacteria. And so this in turn causes the formation of a shallow soil on the sand with an increase in organic humus content. Now at this point you can start to find rabbits in abundance on these dunes, where burrowing is relatively easy because the dunes again have been stabilized. So in some cases the burrowing has been found to destabilize areas of the dune, and this changes the composition of the locally growing plant species. But in turn the rabbit droppings will then increase the humus content of that growing soil layer. Then you will find nesting gulls, improving the nitrogen content of the soil, and also invertebrate species which will inhabit the yellow dunes, including things like snails and also insects. So now that we have an increasing biodiversity here, because the stability of the dunes increases and the soil quality is improving, and so the environment is becoming less harsh and other species begin to replace the marram grass. So we have an increase in diversity on moving inland from the yellow mobile dunes to the fixed grey and brown dunes. So species diversity changes from a few specially adapted organisms at the start of dune formation to a complex community inhabiting the older and more established dunes. So on acidic soils, the yellow-grey dune community that develops is typically a heathland community. Heather, gorse, willow, lichens and mosses will thrive in this more sheltered and stable environment. So here we see gorse, which is present on the Dawlish Warren on the dunes at the Dawlish Warren in Devon. So at each stage, therefore, there is a gradual replacement of one community with another as each group of organisms exert changes on that environment. So this view of older dunes with a more diverse community again is at Dawlish Warren in Devon. Again, these changes will continue until you've formed a stable community and no further changes in the community composition can take place. So this is what we refer to as the climax community. Now, the sequence of events where the organisms bring about changes in their environment such that one community is gradually replaced by another, again, this is referred to as ecological succession, and each community in the succession is called a seer. So we see that there's been an increase in species diversity from the sparsely populated, unstable dunes at the beginning of the succession to the fixed, more stable dune community populated, as you can see here, by the shrubs and the flowering plants. Now, the stable climax community that develops over many years is usually a woodland community. So, over many hundreds of years, the accumulation of organic matter in the soil and the favourable changes in other environmental conditions will support the complex woodland communities. So here we see a view of Dawlish Warren in Devon, and this complex woodland community ultimately developed by succession from a dune, from unoccupied sand dune communities. So abiotic factors will dominate in determining which species can colonise that initial harsh environment at the beginning of succession. But then, later on, biotic factors such as competition between species, these become increasingly important in determining which organisms ultimately will colonise the habitat as the succession proceeds. Now, the changes of primary succession can also be observed in ponds and lakes where vegetation along the shallow edges may grow further and further out, which ultimately create marsh or dry land. So young lakes are oligotrophic or little nourished because they contain few nutrients. These sorts of lakes will support relatively few organisms. Now, oligotrophic lakes will eventually begin to fill with sediment as water runs off from the land. The sediment provides nutrients which allows the lake to support a greater diversity of organisms. So nutrient-rich lakes are described as being eutrophic or fully nourished. So the continuous input of sediment ultimately brings organic matter and nutrients to the boundaries of the lake, and therefore the shores become shallower and start to be colonized by plants which make use of that organic material. So in turn, as the plants become rooted in the sediment, they tend to accumulate more sediment. So the water becomes even more shallow and starts to become able to support marsh plants. So the sediment runs off into the lake or the pond, enabling the shallow roots of submerged plants to anchor themselves, and then the sediment accumulates. So once that accumulates and the plants start to root themselves, the humus content will increase because those plants eventually will die and decompose, and this adds to the organic humus content of the soil, in turn making that soil more fertile. So the enriched sediment allows for the growth of deeper rooted plants, which in turn will accumulate more sediment, contribute more organic matter as they die, and this process of succession, and in some cases climaxes with the growth of the woodland. So secondary succession on this abandoned agricultural field takes about 120 years to develop from the pioneer annual plants to the climax woodland community. So in general, secondary succession tends to be completed in much less time than primary succession, and the reason for this is that the soil is already present, and also there may be surviving seeds together with seeds from neighbouring communities which can quickly colonise this area. Now, the clearing of areas of forest by the slash and burn technique leads to very rapid secondary succession. So in this example, the growth of new forest during secondary succession produces a habitat which is very different from the original. Secondary forests tend to display denser growth and contain species not found in the original primary forest. So we'll finish off with this table here, which I will leave up on the screen for a little while here. This table basically summarises the features of early stages versus late stages of succession, and I would recommend that you guys familiarise yourself with these, but this is basically a summary of what we've been talking about here. Okay everyone, well we've reached the end of the third hour and we've actually managed to complete our coverage of all of the topics that we said we were going to cover, so I'm pretty happy with that. It means that I don't need to film another video for this particular webinar. I hope that was useful to you all. I see that we have a bunch of questions in the Q&A box here, so I will sign off from the YouTube live stream and I will start to address those questions in turn. But again, if any of the students in attendance do come up with any further questions, you will have access to the course module on this topic on my website, and you can post your questions either in your private coaching area or in the comments sections for each individual lesson page. Alternatively, those of you who are registered students on my site, you can always contact me privately via the social network on the site. You can find me on the student directory. Those of you watching this as a recording on the YouTube channel, please do by all means type your questions into the comments on the YouTube pages for this webinar, and I will do my best to address your questions there. So once again, everyone, thank you for attending. I hope that was useful, and I will see you in the next webinar. The next one will be tomorrow, and it is on the subject of, let me just see here, I believe it's on the subject of the control of gene expression. That again will be at 11 AM tomorrow, and I will see you during that webinar. I hope that was useful, and I'll see you then. Take care.